Okay, good afternoon, everyone. So we're going to kick off this session. So firstly, who I am, my name is Cooney. I'm an R&D manager in Boston Scientific, Clonmel, working in urology and pelvic health. I have many members of my team here with me this week, and our visiting director, Rob Lund, who you're going to hear from later on. It's a great honor for us to be sponsors of this research week for the Bernal Institute. Once this works. We've just spent the morning kind of reviewing some of our existing collaborations that we have ongoing with many of you, the members here in the Bernal Institute. I suppose it was a really invigorating morning to seeing the output of the work and how it relates to what we're doing in our current research in urology. So I suppose just briefly, like why that is tangible to us and the products that we innovate. In terms of who we are for Boston Scientific, we are a company who is dedicated to transforming lives through innovative medical solutions, which ultimately improve the health of patients around the world. So the patient is at the core of what we do, and addressing their unmet needs and improving their scenarios is why we exist and why we have our core values, which you see on the right-hand side of the slide, which is the backbone of what we do on a daily basis. I suppose just this slide here probably hits home what we do across all of Boston Scientific. I think just the top left tells the full story, where we treat 30 million patients every year, which is a really, really powerful message when we think back of everything you're doing when you're analyzing data, running an experiment, and you know, going through difficult times in all aspects of what you do. This is what brings us back home to us. I suppose across that we have over 17,000 products which enable that amount of treatment. And I suppose then as we look forward, right, so every year we invest approximately $1 billion in our R&D that will enable that next generation and on the innovative products that we have. And hence it's how we use that and how we collaborate with the likes of everybody here in the Bernal, in the University of Limerick, which allows us to accelerate the work of what we do and ultimately bring those new products to market quicker for our patients. The area that we are focused on is urology and pelvic health, which is a big division and a big focus for Boston Scientific. And that's in probably 4 key areas, right? So it's in the areas of stone management. So you can see the graphics there in terms of the number of percentage of the world's population that will be affected by kidney stones. We have prostate health. Again, so a very debilitating disease that a large number of men will suffer from. Where our focus is, and Rob's focus, which you hear from later, is in the area of prosthetic urology. So it's around the area of ED. And then we also have the female pelvic floor management as part of our portfolio as well. So we treat a wide range of urology and pelvic health needs for the patient population. So I suppose as we look back then and to the work we do with you, I'll call it out now, the future talent that we hope to collaborate and bring into Boston Scientific, this is why we do what we do. So there is unmet needs. We have products that address all these needs, but we have an innovation pipeline that we love to accelerate further. So I suppose that just sets the scene of who we are and what we do. And what I'd like to do now is introduce the speakers that we are collaborating with on our academic partnerships. So first up is Eric and Valeria, who are going to talk to us on IPP and their future work. So thanks a million, and please do ask questions of everybody and how it relates to the work that we do.
Hi everyone, I'm Valeria Nico. I'm a research fellow in the Bernal Institute. So I'm gonna present you a case study we did for Boston Scientific in terms of addressing 3 key aspects of the development of a completely new generation of implantable devices. So we did the project in collaboration with Boston Scientific and an Enterprise Ireland project So we analyze three main aspects: corrosion testing, dielectric breakdown, and nano-leakage. So we are dealing with new type of implantable devices. This particular one is an active implantable. So it's the first time actually there will be an actuation in the system and in implantable devices, and there will be fluid moving around. So it's very important to understand why corrosion can happen, if can happen, and leakage. Obviously, we are dealing with fluids, why we are interested in leakage, and then we'll see why they break down. So as part of corrosion testing, one of the worries was that we have metal foils that are moving. There is an applied constant voltage on those foils, and we have saline fluid. So even if the metal foil is made up of titanium, there are laser weldings on it, and one of the worries is that the laser welds behaves like a different type of material, so a different type of metal. So the worry was that there could be potential galvanic corrosion between the normal grade 5 titanium and the laser welded. So it's important in this case to study if corrosion can happen, because we need to predict the impact of corrosion and how this affects the life of the device. So we carried out electrochemical testing using a potentiostat, and also we analyzed, we imaged the sample with our scanning electron microscopy to detect corrosion, and we tried to accelerate the testing, tried different pH, so to try to actually cause corrosion, but we were not able to observe any corrosion at all. The second test was the electric breakdown. So these metal foils are the interface between the fluids that are flowing in the device and all the electronics. But obviously it's a metal. That means that it's not insulating the fluid with the electronics. For this reason, there will be an insulating layer to actually insulate the fluid to the electronics. So it's key to understand if this thin insulation layer will degrade, will break, what's going to happen under stress conditions and high temperatures. So we tested with higher applied static voltages and at 37 degrees. And we tested both standard commercial dielectric materials and then thin epoxy films, because one of the possibilities to make life easier when assembling the device was to replace actually the dielectric layer with a thin film of epoxy to make assembly easier. But we actually didn't detect any degradation of the normal dielectric materials, but we had 4 samples of the epoxy films, and both samples failed within a few months, short-circuiting the electrodes. So it was kind of interesting for us to see how definitely it's not a good idea to use the epoxy as insulation layer. Finally, the last test was to detect nano-leakage. So in the case of corrosion, so in the case small holes are forming in the device, how much liquid are we losing and how much the device will last for. So one of the key issues, this was probably the most challenging test. The key issue is that current leakage testing are done with gases. So leakage is used using, for example, helium testing. And this is not good for us because it doesn't give us the correct sensitivities. In the device, we have a liquid to air interface. So we have a liquid leaking into an air gap. So we had to come up with a completely novel testing solution that allows us actually to predict the leakage rate depending on the hole size. So this is just to give you an idea of our, like a case study, so what we did, what the type of testing we did to develop the new generation of implantable devices, in particular for urology, and Eric will give you a broader view of Stokes lab capabilities.
Thank you, Valeria. So hello, everyone. So a lot of this work that we've done with Boston was enabled by essentially the team in Stokes that could collaborate very well with Boston. Stokes, which is under Bernal, is essentially a multidisciplinary group. We kind of focus on microfluidics, mass transport. We do aerodynamics, genetic, and biofluidics as well. And our application areas, we look towards, biopharma, manufacturing, aeronautical, and energy. But all of these things, I just wanted to give you a flavor of what we do in Stokes, because each of even the things that we do can be used in urology. So in the previous IPP study, we were using material testing, reliability, and novel fluidics to measure leak detection. So all of these things are disparate, but you can come together very well for a project. So one of our key areas is material studies. This is kind of primarily driven by Dr. Morris Collins. So he's producing, for example, carbon fiber from sustainable materials. Sustainable materials. With Fernanda, he is also producing immunoprotection using hydrogels, hyaluronic acid. And we also do characterization. So we went and we did at the diamond synchrotron how material deformation changes the crystal structure in implantable devices. So this is polymer devices looking at the crystal structure with basically a gamma laser. These are fundamental material characterization stuff that we can do. Again, we look at thermals, which is important. So myself and Valeria developed like micro pumps, which was initially under Noel Smith. We were looking at implantables, but magnetics didn't like to be in the body too much. But this has gone on to, we're looking at this for satellite cooling. So we're just finishing off a projects with ESA on that. Valeria's energy harvesting. Jeff is also looking, so stuff like PIV, as in how fluid moves in confined spaces. So this is jet, rotational, and this is experimental data using PIV techniques. Again, really interesting, how does fluid actually move through a system? Reliability, as in we've had a long history in that. Corrosion, galvanic, we use mixed flowing gas, flowers of sulfur. Accelerated ATC testing, so thermals basically heating up, cooling down. And we also do like creep mechanical fatigue testing, so essentially moving things. And these are example projects that we've done. IP generation, so we have, in terms of microfluidics and optics, liquid bridge mixers, drop-off junctions of fluidics, and you can see here that we developed high-density arrays for PCR. I think everybody knows what PCR is now. Don't have to explain that. And then also system integration. So taking all of these things together, we built with a spin-out company, our own spin-out company, high-throughput PCR system. So this particular one produced over 10,000 individual PCR experiments per hour, several thousand PCR. And we've moved on to generation 2, which is high-throughput T-cell response detection for immunotherapies. So all of these disparate things, interdisciplinary, which it does matter, come together, and you can actually do something very interesting and produce and deliver on projects, essentially. So, thank you. Yes.
Of course, yeah. Oh, yes, sorry.
Hello.
Oh, you're on. You're good.
So, Eric, you mentioned there a few minutes ago about the pumps, and yes, indeed, MRI probably would have been a challenge.
Yeah.
But an absolutely amazing technology, totally novel. You know, we were so impressed. I mean, I think, Rob, you saw them early on as well. Really impressive, very efficient. A couple of questions. What's the potential for incorporating valving? And secondly, do you think there's a commercial path? I think there is, ultimately, because it is so novel and so effective.
So, yeah, we love this technology, and we were like devastated with Boston. But in terms of this is going forward, we're obviously pushing on for this. And so this went for testing with ESA to do essentially with higher density energy satellites, so something like the CubeSat and even larger, that their energy densities are just increasing and heat pipes are no longer doing it. So they're looking towards pumping. And one of the issues, and most efficient, is you need to do it with pretty nasty gases, ammonia, at high pressure, so it's a liquid, so two-phase. And that also says that you have to run it at 60 bar. So we've tested these. These pumps will quite happily, with a static pressure of up to 200 bar, and they're immune to ammonia. So they're also, we did vibrational testing, micro-vibrational testing, thermals, we did a lot with ESA. And so that was very, very positive. It passed with flying colours. And so we're doing the next, talking with companies about the next set. So they're looking to do, it should be, hopefully, early next year, they want a test bed for, like we did for ESA, it's an array, so it's 5 pumps in parallel, an array to prove the technology to them. Not so much prove they believe us, as in they want to play with it, and so we're going to build them that. And so it's looking very bright for it. And in terms of, sorry to get back to the valving, we have done valving, latch valves in particular. I remember we were talking about that, but the leak rates, we didn't really concentrate on that very much, as in it's something we can do and come back to, because when we were doing it, we were kind of very much concentrating on ultra reliability. And so we stepped away from something like, we were like silicon nitride, that was our material of choice. But if we can change that to, more of a silicone type of sealant, we can do it. We can. It's in the same, I think you've seen a variation. So yes, we can do it. It's in the leak rates, 4 microliters per minute. We'll have to talk about that one. Even measuring that one isn't that much fun, as you well know. Yeah.
No, thanks a lot, Eric. That's great. Appreciate it.
There we go.
What is the range of the particle size that can be used in the pump?
So it would be quite happy to take up to maybe 200 micron, high density. It won't take a slurry. If you try to put a slurry through it, what happens is that it won't stop, but you'll just have a backload of slurry. But low density of particles, it still will just run happily. So up to 200, smaller makes no difference. We have it, as in, we've tried it with particles, it's fine. But with, as I said, with a slurry, not so, it'll run, but it'll end up giving you the fluid and just a backup of particles, I would imagine, up on the other end. It all depends on your density of particles.
Thank you.
Thank you all.
Great, so thank you very much, Eric and Valeria. So next I'm going to welcome Sarah Guerin, who's going to talk piezo, current and future.
Okay, folks, so Valeria and Eric very much working on the current electromechanical research that's going on in Boston Scientific. In my group, which is the Actuate Lab here in the University of Limerick, we're looking towards the future of piezoelectric sensing and how that can be integrated into urological devices in Boston Scientific. So firstly, I apologise to all the Bernal people in the room, but just to define piezoelectricity, they're an ubiquitous part of medical devices across the board. They interconvert electrical and mechanical energy, so they're sensors and actuators. It is ironic that they're a huge part of medical device technologies around the world, but they're also the last remaining mainstream technology that actually contains a lead-based piezo ceramic. So our goal in my lab is to develop eco-friendly alternatives to these materials. So here's just a list of how ubiquitous it is in medical device technology. Today we're going to be talking mostly towards driving these towards pump applications, but they're a huge part of ultrasonics, of cardio. There's a huge future for them in self-powered implantables and self-powered medical devices. So what we want to do is we want to use biomolecular crystals. We want to phase out these lead-based materials. Hopefully we have funding until about 2026, 2028. And we want to phase out the toxic materials that are currently used for these applications with these eco-friendly alternatives. We've already shown that they have very favourable electronic properties, dielectric constants, so we've shown that they're highly efficient energy harvesters, which means that they could also be used to harvest energy from inside the body to power certain medical devices. An exciting thing about them is we can also tailor their electrical properties for specific medical device applications based on the chemistry of the materials themselves. And I can see my two chemists sitting in the middle there who totally agree with me that that can be done. So the phrase in our lab is getting the lead out. It's not just about developing a completely new class of piezoelectric material. It's also about making that material versatile enough for the thousands of applications that piezoelectric sensors are used in and the hundreds of medical device applications. So we started off our work with Boston Scientific Clonmel by benchmarking certain characterization methods on their piezo ceramics that they're currently using, but we need our materials, and we've gotten to the point where we can make our materials in the variety of different shapes, sizes, thicknesses that we can do with ceramic-based materials so that they can be very versatile and easily switched out for the applications that ceramics are currently being used in. So what we can do with our materials is we can mold them into various sizes and shapes, and again, depending on the chemistry of the molecule that's used, we can tailor the actual properties that we want, such as dielectric properties, mechanical properties, and all of those things. So in terms of what we've done already, in terms of showing that this material, which generally when you hear biomaterials, you think of soft, you think of squishy, those kinds of properties. So we've been working to show that these materials, such as amino acids, really small biomolecules, have technologically interesting and technologically viable material properties. So what you can see here is purely in terms of sensitivity, so the amount of output voltage that you get per input force or input vibration. We're already outperforming a lot of the current polymer-based piezoelectric, so this is our glycine sensor that was validated with a team in UCD. And a big, as I'll mention, another benefit of them is they're about 1,000 times cheaper than piezo ceramics. So we can easily test these materials, scale them up, and have them in very large-scale arrays depending on the application that we need, and very high mechanical strength, durability, and life cycle for these materials. So what we're currently trying to do in my group to really advance this technology is we're tackling it on 3 different pillars. We know all the things that we like about these piezoelectric materials and this new class of piezoelectric materials. But the first thing we need to do is make sure that our input-output is performing how we want it, so increasing our piezoelectric strain constants. Our group has a lot of expertise from materials modelling through to the crystallographers that make these crystals and then through to the device engineers that can move these towards medical device applications. So we're both increasing their performance, controlling how they grow, as you saw on the previous slide, and now what we want to do is move these towards specific application-driven medical devices. So compared to other ceramics, other polymer materials, it's a very low cost, which is beneficial when you're trying to really change a whole class of material that's used for a specific application. So just to run the numbers on it, for ceramics, we have barium titanate, PZT, niobium pentoxide. All of these raw materials are very, very costly to us, to the people purchasing them, as well as highly damaging to the environment. So our materials are not only eco-friendly and high-performing, but very low cost for medical devices, which feeds back into a lot of the goals that we have working with medical devices towards patient cost and patient experience of these medical devices. So this is to show how we've moved in the past 3 to 5 years from very basic proof of concept type devices back in 2018, where we were taking our crystals, placing them between electrodes and just showing that they had this phenomena, to quite high performing. This is Sai, a student who worked in our group for the past couple of months, who was able to make an actual mini Fitbit monitor that could detect her heart rate, detect her breathing, and detect a lot of physiological signals. So we've really gone from our proof of concept that these materials have this phenomena and that they work, to nice demonstrators for the future they have in medical device technology. So our outlook in terms of our own research, and particularly if we can go down the urology and the medical device side of things, is piezoelectrics that can be modelled to a very high accuracy. This means we can screen biomolecular crystals and tailor them to specific patient needs or specific device needs even. But what we can do is we can understand at the atomic scale what makes these materials piezoelectric, and that allows us to tailor design them in a very unique way compared to ceramics or polymers or other materials. And our modelling has advanced to the point that we can even screen at what forces these things are going to break under, how they're going to behave under certain stresses and strains before we even grow them in the lab. So there's a nice workflow from our modelling to our actual material development, all the way through to working with Stokes and Eric and Valeria in the future towards integrating this into these kinds of technologies. So in the future, we'll have our atomic scale toolbox for how to engineer eco-friendly piezoelectrics for different medical device applications. That will allow us to combine what we do with our modelling and what we do with all of our different screening criteria and all the various electromechanical properties that come together for a piezoelectric medical device. And we can combine that with our advanced characterization here in the Bernal, here in Stokes, to really advance these. And hopefully, with working with Boston Scientific, we can help them to become the first company that embraces these low-cost, eco-friendly sensors to really to really be the first people to advance the uptake and show how effective they can be, both for us as scientists, for Boston as manufacturers, and for the patients. So thank you.
Thanks very much, Sarah. So questions for Sarah? I'm sure there's lots.
Hi. Hello. Can you hear me? Yeah, so you said you can manufacture materials that are 1,000 times cheaper than PZT, but regards material properties and performance, how far you are from PZT, pretty much?
Yeah, that's a great question. So I suppose commercial PZT, as you know, about 350 to 550 picocoulombs per Newton for commercial applications. Our current record for high performance is about 180. So with the correct order of magnitude, it's just about hopefully tweaking the chemistry either through discovering a particular crystal through the modelling or through engineering the growth mechanisms to maximise that response. So correct order of magnitude, but work to be done.
Far away anyway.
Yeah, Sean, can we just lock in the pricing now?
Good idea.
I suppose just like probably what would concern is how do they break, and would you anticipate the same kind of failure mechanisms, or are they different? So for example, what we see from electrodes, or we see the effects of humidity and voltage, or are they similar or are they different that way?
Yeah, they have, I suppose they have superior mechanical properties to current ceramics. They're much less brittle. We've cycled them through for other applications in terms of the life cycle analysis. But we have to package these things very carefully. I mean, they're biomolecular crystals, especially if we're going down the implant route. We have the raw material that works now, but we're currently working on developing sealing, packaging, insulation procedures to protect them from anything that they mightn't be able for.
Just before you were saying you were growing them on a substrate, so the adhesion to the substrate, what kind of mechanism is that that's getting it adhered?
Yeah, so it highly depends on the metal. So we see ourselves, even the difference between aluminium and copper is huge. They really like growing on copper. There's little reactions that take place that form other kind of subproducts that really keep them on the copper substrate for those applications. But for the ceramic-dominated applications, we'd rather grow them without a substrate, so that technique that you saw there was just the crystalline element, and then to sputter them after that to make your electrical contact. So we're cutting adhesion out of the equation altogether that way.
Any more questions for Sarah? Oh. It's going to be a tough one now.
This has been building for a couple of presentations now, Sarah. I always think about the mechanics of it, and I wonder, is there a preferential way, like, of deforming the material to increase the electrical activity, like shear, compression, extension, mixed mode? Is there some way of doing it that you can maximize your output?
Yeah, that's a great question. Thanks, Owen. So the response is highly anisotropic. So for every, if you look at it at single crystal level, there's up to 18 different responses. So you can have your, exactly that, you can have your transverse, you can have your shear, you can have your longitudinal forces. And what's great about the models is it can screen that for us as well. So the models tell us what direction we should orientate the crystals along and what force we should apply to get the most output. Yeah.
Okay, so you can kind of tailor to the application based on that. I imagine compression is usually the easiest thing to kind of achieve.
Yeah, exactly. What we like to see is what's called a D33 constant. So you can apply those tapping devices where you're applying your force along your Z-axis of your electrodes and getting your charge out. When you start to introduce shear, it's only if we need it for specific applications. For the ceramics, it's generally that, yeah, compression longitudinal interaction.
Great, thank you, Sarah.
Great. Any more questions? Perfect. Sarah, thank you very much. So next I'd like to welcome John Mulvihill and Shaza to talk about coatings and the future.
Right. Afternoon, everyone. So my name is John Mulvihill. I'm a senior lecturer in biomedical engineering and course director. So with that, I like to always try and engage people. So you've been talked at for a little bit, so I'm going to flip it to you. So I just have a question here, and you can use this QR code to answer it if you want. So the question is, what percentage of women will experience a urinary tract infection in their lifetime? So do you believe it's 10%, 20%, or 60%? Now, I'm going to answer that question at the end of this talk, so you can see why I'm doing this, to keep you engaged. But this is related to what I'm going to talk to you all about today, which is urology research in the Bernal Institute, but specifically in the Bernal Biolabs. So I'll just give you a second to take that picture, but I'll put this up again later on at the end of the slide. So I'm here to advertise not just myself, but our group here, which is the Bernal Biolabs. And it's that red square, or rectangle, should I say, that you see here. So we're just here on the second floor and the third floor here. So our lab is just behind you all here. I'm going to show you what techniques we have and how we can apply that to urological research here in the Bernal. So and just to advertise how good we are and how sustainable we are, I just want to show our My Green Lab certification. So that just is about the practices that we have within our lab. And myself and Dr. Andreas Graubacher started in 2020. And we achieved the second highest score in the whole world for My Green Lab certification. So what that means is what we do in there is to make sure that we do things sustainably and that we're not wasting energy and items within there. So it just shows you our mentality that for things like urological research, that we do it in such a sustainable manner. So this is the different people involved, or the PIs within the Bernal Biolabs, and on the right side here you can see the type of research that we carry out and the different areas for all our different colleagues here. But today really I want to talk about urology and the techniques that we can apply to it. So this is a very busy slide, and the idea is to show you more the techniques that we have, and I will zoom in on different ones and show you how they relate to the to urological research, particularly the ones on the top there. So the question is, how can we apply these techniques to achieve patient outcome in urological research? So we all know about medical implants, and when we think about implants, we probably think about orthopedic, right? So our bones. So you probably think of hip replacements, knee replacements, even elbow replacements, and even cardiovascular, stents all throughout the body, aortic valves, and pacemakers. So I'm just going to talk and give you an example of one of these and how we can improve upon them. And to be relative to today, urology. So it can be implants, penile implants, as well as urinary catheters, so indwelling urinary catheters. And what's the commonality? Why did I list out all of those? Well, the biggest issue with them is bacteria. So when they're implanted, we want to ensure that they're implanted in a sterile way, but that cannot always be guaranteed. I mean, no matter what we do, we know with COVID, we cleaned our hands and we did everything we could possible, but it still went around the world, albeit it was a virus. But even in the surgical theatre, we believe to be completely clean, there's still a chance that bacteria can get onto an implant, and that can cause huge problems. So what problems do I mean? Is that bacteria can live on the surface of your implant, and it can grow in the body, and it will develop some sort of slime or biofilm that can create this protective layer around the implant, protecting the bacteria, and ultimately, no matter how much antibiotics that you put onto that and to that implant or try and remove the bacteria, it won't get to it. So how do we do that? So looking at, let's say, specifically a pacemaker, which is a very critical device, what can we do? So if we look at the cost of this alone, so there's a risk of death increases threefold with infection within the first year. There's a 19 to 18 day average time in hospitals for each infection, regardless of what type of implant it is, and the costs as well to the hospital itself. So it is a problem no matter what type of implant, from cardiovascular to orthopedic or urological. And I'm just going to show you here, the concept is that why don't we put some sort of protective layer around it, and just using a pacemaker as an example here. But we are now in an economy, and I showed you our sustainable practices. Adding materials is something that we shouldn't be doing, right? So we have a circular economy bill that is starting now here in Ireland, and we don't want to be adding materials. We want to be working with what we have and making sure it does the job correctly. So how can these techniques help? Well, what we can do is look at typical materials that are implanted in the body, be it titanium, be it polyurethane, be it copper, whatever it is, and see how it engages and interacts with the body. So can it naturally kill bacteria? Can we do something to the surface to alter that, to ensure that it kills the bacteria but doesn't kill the cells within? And that's what we can do in the Bernal Biolab. So we can take different types of materials, put different coatings or shapes on them, and see what effect it has on the bacteria, which we want to get rid of, or the cells, which of course we don't want to harm. So be it a stent, be it a catheter, be it even a pacemaker, we can choose the type of material that we want to investigate. But I'm just going to specifically look at urology, and what makes us different here is that we can mimic the in vivo environment. So what does that mean? We can try and mimic what's going on physically and cellularly within the urethra. So in this case here, you see the catheter implanted within the bladder and the urethra itself, and just showing that bacteria, if there is presence of it, will build and grow over time. So what can we do? We can mimic this environment and understand how the bacteria grows. And just on the right here, you're seeing the in vitro side of it. So we can mimic the bacteria. We can mimic the cells within the urethra itself and understand, is it growing? Does this material encourage or discourage any growth of that bacteria? And what I'm going to show you here is a slide of a urethra. And it just shows you what we can mimic on a kind of cellular and structural level. So this is of a porcine urethra, and what we can do is recapitulate and mimic this environment and deploy an actual implant in it to see how it works. So it just shows you the kind of detail that we can carry out. And further to that, we can mimic the environment. So in the urethra, in the bladder, it's not a static environment. There is movement, specifically voiding your bladder of urine. So that's going to cause some sort of mechanical shearing. So it's not just mimicking the cellular or structural environment. We can mimic the actual flow and mechanics itself. And we can do that in vitro. So now you can come along, design your product, design your material in a real environment without having to use human tissue. So beyond that, we can look at the mechanical characterization as well. So how is your implant affected by it, or how is that tissue affected by your implant as well? And just looking here, just to give you a sense here, but it's very difficult to see, but that is cells on a substrate, and that probe there, you can actually isolate a particular cell or bacteria or whatever it is that you're investigating and get the mechanics of it on a cellular level. And we can visualize it even in its environment. So you're seeing there images of, in this case, cells. And we can bring it up to the ex vivo level, so not just the in vitro or cellular level. We can bring it up to, let's say, a model of the urethra, a porcine urethra in this case, implant your device, introduce bacteria and see the effect that it has on the implant itself, the material you're investigating, or the tissue itself. And this is just to show you the kind of results and capabilities of it. So we can mimic here on the top left, this is the setup that we create. We can look at the structure and the effect that it has on the top right. And on the bottom here, we can get an idea of the mechanics and the changes to the tissue in this case because of a particular implant. So what does this mean? So how can we apply this to patient outcomes? By understanding the effect of the bacteria or the type of device that you have, by using this animal or in vitro testing, we can design and redesign these types of implants, the surfaces that we put on them, the coatings that we put on them to try and get and achieve patient outcome. So with that, what I promised you is to answer this question, which is, again, what percentage of women will experience UTIs in their lifetime? And the answer is 60%. So it's quite high. And you may be wondering, well, how about men? Well, the answer is around A, so 10 to 13% for men. So there is a difference, and when we talk about urology research, we need to understand the differences in the anatomy between male and female. And it's not just gender, but also age. So you can see there on the bottom right, it increases as we age. So urological research is incredibly important. It's something that we should always actively be looking at, be it gender and age. So with that, thank you for your time. Cheers. So I'm going to hand you over to Dr. Shazza Darwish, who's just going to talk about a little project that we have with Boston. So, over to you.
Yeah, I will talk about a fellowship that we applied to with Boston Scientific, which is the SFI Industry RD&I Fellowship. First, I'll start by introducing myself. I have a BS in chemistry and B in chemical engineering from American University of Beirut. After that, I did my PhD in the process engineering group, and I worked on the manufacturing of pharmaceutical co-crystals and metal organic materials. Followed by that, I worked in the crystal. I did 2 postdocs, one in the crystal engineering group where I worked on atmospheric water harvesting and green synthesis of metal organic materials, as well as now I'm working in the pharmaceutical manufacturing center and working on sensors for developing a sensor for powder blockage. Throughout all this, I have accumulated experience in manufacturing and processing, material science and characterization, and this was developed by using all the facilities here at Bernal, as Dr. John talked about, and such as PXRD, TGA, FTIR, and I also worked with industrial partners in which I also filed invention disclosure and I have more than 20 publications in these respective areas. So I would like to give an overview about this fellowship program, which is funded by SFI, and it seeks to support the academia-industry interactions and will be an important vehicle to support the industrial research, development, and innovation. The grants in this program will be given to researchers that wish to spend some time in the industry and have experience in industry. This program will be beneficial for both industrial and academic partners, and as outlined by its objectives, which are to maximize the economic and societal impact in Irish industries, to foster the competition advantage and future resilience by engaging in Irish research, by engaging in industrial research. It will improve the employability prospects of the researchers, as well as it will support the exchange of knowledge and expertise between industries and academia. So in particular, I collaborated to apply to a fellowship with Boston Scientific about a stable interface for smart implants in the body. As John mentioned before, that bacteria can adhere to medical devices implants and prevent antibiotics to reach their surface. And in this fellowship, we will mainly aim to study a unique interface for the implants that fights infections and can be used across all medical devices, thus offering fit for all solution. And it will be divided into 4 work packages. In the first work package, I'll be introduced to Boston Scientific, get training, and do some literature review. The second one will be mainly to characterize some coupons made that are used to make implants, and to study their surface analysis and testing. And finally, in work package 3 and 4, I'll do some in vitro and in vivo testing for the interface, respectively. Yeah, thank you.
Thank you, Shaza. So I suppose any questions for John or Shaza on what you've just seen? Tom.
Hi, Tom Sinnott here from Boston Scientific. It's more of a general question, I suppose, about we're learning a lot from your research, you know, helping knowledge gaps in our business. I suppose, is there anything that you've learned from working with industry and feeds back into your research?
That's a good question. You don't have to do as much teaching as we do. That's the main thing. Yeah, it's just standard of operations, procedures. You know, I know Shaza's hopefully going to be working with you in the next year, and just seeing how you carry out your protocols and actually trying to implement that type of procedure within our own facilities here. Because you saw we've a very large lab and lots of different things going on. So to try and implement those procedures and standards of operations, because it's just us trying to implement this. So that would be the main thing, which is a very vague answer to it. Behind you there. Yeah.
Yeah. Hey, is this on? Okay.
Yeah.
Quick question for you. You know, as you think about infection models, you know, they're quite challenging to do because either, you know, especially in an animal where you either get like this raging purulent infection, or the animal tends to just cure up the infection all on its own. I think one of the things that's of interest to industry is what I'd call these subclinical infections, right, where there's a biofilm formation, but it's not necessarily manifesting itself into a full infection that a patient can detect. Do you guys have techniques and stuff that you guys are working on developing for something between a raging purulent infection and a subthreshold biofilm?
Yeah, that's a good question. So there are standard models of creating in vitro biofilms that we'll probably work off of. But yeah, when you go to the ex vivo level, it'll be hard to determine whether you have a model or not. And that was the point of that final work package there, is just creating the model in of itself would be an achievement of its own. But we have capabilities in keeping ex vivo tissue fresh. So then applying those bacteria to it to see if we can just see, does it increase over time? So just using basic CFUs and determining has that increased with or without the interaction of those materials. So we'll just use our in vitro model first to determine whether we're getting that kind of growth. From there, we'll decide, okay, this is a good material that we're going to work with, and then kind of raise it to the ex vivo level. So that, because it's one thing using a coupon, right? It's another thing putting it on the actual material with all the geometries there, and that was the point of the ex vivo. Set it up to see, okay, well, this is a real implant. How does it engage with a real tissue? So yeah, I mean, creating those bacterial models will be in itself a project probably. So, good question.
I suppose generally, just how do you see the landscape or appetite in industry for antimicrobials at the moment? Has it dropped off, or is it growing? There was quite a trend back in 2014, 13 that everyone was hunting for one. Has that tailed off, or is it still fairly active?
I know myself from the area of urological research and even in industry, it's increased, right? But that's the vague urology. I know myself for even UTI diagnostics, that has increased incredibly in even the past 3 or 4 years. So whether it's a cure or a diagnosis, in terms of an antimicrobial surface, yeah, I agree. I'm not too sure about what direction research is going, but we can certainly look at the material level. So instead of adding another material, could we look at the surface that you're given and see if even altering that surface can have an impact on bacteria. But in terms of, is it increasing? I'm not too sure. So, yeah.
Okay, any further questions for John or Shaza? Perfect.
Apologies if I missed this in the presentation, but I'm just curious about the in vitro side of things. What cells, or like, is it more than one? Are you looking at inflammatory markers? Just getting back to your kind of subclinical type infection, or?
It'll be a basic viability assay. So for this particular project, it's cardiovascular, so we'll look at endothelial cells. Does it kill them or not? It'll be very basic for what we're trying to achieve. So yeah, endothelial cells more than likely with just cell viability. So no particular markers in that case. It's just, do they like the presence of it or not? So we do have a cell impedance platform, and that can look at the growth of the cells live. So instead of it being an endpoint, after 24 hours they're dead, we can actually see how they're being affected by the presence of this material literally every second. So that's what we're going to do, a combination of viability and cell impedance on that particular cell. But what's great about this and what we're trying to design here is it's just not for, in this case, let's say a cardiovascular implant. It can be applied to urological and orthopedic. So what we hope to have here is something that we can keep applying to other materials.
Okay, anything else? Great, so thanks again to John and Shaza. So now I'd like to welcome Eoghan and Olwyn to talk biomedics and oncology.
Yes, hello everyone. My name is indeed Eoghan Cunnane. I'm a lecturer here at the University of Limerick. And what do I do here? So what I'm funded to do, regardless of what I actually do, what I am funded to do is to do with broadly, I suppose, population trends, and more specifically to do with male infertility. So if you if you look at the global trends, if you look at birth rates, you can see that there is an absolute contraction of birth rates across the globe, and this is due to a lot of factors that a physical scientist should probably not concern themselves with, because it's to do with lots of socioeconomic things that certainly I did not want to tackle in the grant. But if we look at this, and this is something we decide is outside of our control, we should decide that we do want to preserve the ability of willing couples to conceive, because that will further compound this contraction of birth rates. So if we look at how we're doing in terms of this, how are we doing in supporting people who want to conceive naturally? We're not doing a very good job, because male sperm concentration has dropped quite considerably since records began. And if you project this decline forward a couple of years, it drops to zero. Now, projecting something forward that many years is definitely not something you should do unless you're applying for a grant and looking to get funded. But you get the idea here that there is an issue with for population trends. There is a further issue with male fertility, and when you look at how it's treated, it's not very good. They're empirical therapies, so they're typically hormonal therapies, and they are delivered in a kind of a let's give it and see what happens kind of way. There's no mechanistic understanding as to why they're prescribed. There's no optimization of dosage, duration, or even treatment choice. And when we look at the ways in which the options we have in which to further evaluate, to actually look at the mechanism, to try and optimize dose, duration, treatment, we find that the preclinical models we have are not very representative of the human condition. And typically when you look at an animal model, you will see that it's not very representative of the human condition. In the case of the testes, I feel that it's even more so. The typical model that you would use is a neonatal mouse. And the reason you would use a neonatal mouse is because their germ cells, their testicular cells will grow quite readily, and making it easier then to kind of evaluate treatments and stuff. But I was able to convince reviewers anyway that this is not a good model. and I was given money by the ERC to try and develop a more representative model. And the way in which we want to develop a more representative model is to have better characterization of the native tissue. So what produces sperm? The testes produces sperm. So we want to develop a more representative model of the human testes in order to try and combat the growing incidence of male infertility. And in order to achieve this, we use a tissue characterization approach. So tissue characterization is good for many things, not just for modeling tissue and increasing the accuracy and representativeness of models. It's also very good for providing a baseline. How should tissue look like when it's in a perfect physiological state? And then how does it look like in a pathophysiological state? If you understand both cases, you're better equipped, you're better positioned to develop diagnostics. It's also very good for understanding medical device interactions, understanding how these solid devices interact with the solid tissue. And finally, it's good for engineering tissue, engineering models that can be used for evaluating treatments, such as the treatments available for male infertility. And if we look at the actual models themselves, we see this typical kind of paradigm of preclinical testing, where at the left end closest to me you have 2D monoculture, and at the far end you've got your large animal models, and it's intuitive that at this end it's quite high throughput, at the other end it's physiologically relevant. That doesn't transpose. So 2D cell culture is not very physiologically relevant, and animal models are not very high throughput. And when you're designing, or when you have the freedom to design a new representative model, what do you want? Oh, that's the wrong way, sorry. You want something that is both high throughput and physiologically relevant. And in order to do this, you need to understand the tissue, and that's what I'm getting at with the tissue characterization. So how do we characterize tissue? And again, this is kind of treading ground that John walked there a minute ago. What facilities do we have here? What techniques can we apply to the characterization of tissue, and then to the modeling of tissue also? So we want to understand the physical properties of the tissue. We want to understand the chemical properties, the spatial arrangement of components within that tissue. These are the things that we want to understand in order to develop a proper model of the tissue. And then once we understand this, how can we actually develop a model? Well, we need a material, a base material to act as your in vitro model. You want to be able to fabricate it in such a way that it's representative of the tissue and high throughput, and you want to be able to analyze it. And then at the interaction between these two, you want to be able to look at the cell-material interfaces. So putting cells back in from the native tissue into your new in vitro model, and using this system that you've developed, this representative system, to evaluate treatments. So what can we do here at the Bernal Institute in terms of characterization, and what do we feel is relevant in order to develop a representative preclinical model? Well, we look at molecular transport in the tissue. We look at drug availability in the tissue. So if we have access to tissue that's taken from patients or tissue that's taken from animals, labeling different molecules with fluorophores, and examining how these fluorophores move through the tissue in order to understand how drugs would move through the tissue in vivo. We can also do macro-scale mechanics for medical device interactions, so taking large lumps of tissue, deliberately deforming them to failure, in order to understand how they are likely to interact with a medical device. And then on a more micro scale, understanding how these tissues, these extracellular matrix proteins, how they interact with the cells on a cell scale level. As John said, we can use nanoindentation, so you're finding a very small area of tissue, nanoindenting it, understanding the mechanics on a micro scale, and therefore understanding how devices, or how to better mimic that region. And you can see here this applied to human testes. So this image here was generated by Isabel, who's part of the group. Very excited. Thank you, Isabel. So you can see here the capabilities, kind of what we can do. So there is you can see on the top right there's actual human testes, and that overlay is a map of the stiffness and how it changes across the testes. And we can relate that then to histological images as well, to try and understand what we're trying to mimic and the properties of the tissue that we're trying to mimic. So that's kind of the physical. Moving on to chemical, we have access to all the typical assays that you would use to characterize the chemical composition of something, but further on from that, we have many spectroscopic techniques as well. So we have Raman confocal for understanding the kind of the composition of the tissue. Raman spectroscopy will give you a fingerprint, which will allow you to quantify and identify different components of the tissue. And we also have mass spectroscopy, which kind of allows you for a less biased, kind of more omics style approach to characterizing the chemical composition of the tissue. So with that, I'm going to hand you over to Olwyn Mahon. And just to kind of preface that, Olwyn is the Jim McCarran Senior Research Fellow. This is not Olwyn here on the right. This is Jim McCarran, who, through the UL Foundation, generously donated the money that's funding the project that Olwyn is working on. So, Olwyn.
Thanks. Yeah, I'm hoping there's not much similarity there between the two of us. So as Eoghan said, I'm a research fellow in the lab. So I'm going to focus a little bit more on, I guess, some of the other techniques and the kind of new infrastructure that we have in the lab that allows us to do such specific characterization of these tissues, either healthy or diseased tissues, and then how we use this then to inform model design. So as Eoghan has already highlighted, the need for complex models is really an unmet need at the moment. So having as much information about the characterization of these tissues and these systems allows us then to implement this into our design of our modelling system. So I'm going to talk a little bit about the techniques that we use and the approaches that we use. So we're very lucky in UL that we had new infrastructure that came in as part of the National Spatial Tissue Profiling platform. And this really had a focus on trying to understand the spatial dynamics within tissues, either diseased tissues or healthy tissues. And what this was allowing us to do was use new infrastructure such as multiplex histology and also single-cell sequencing methods. So when we think about tissues, we know that they're very, very complex. And a lot of the techniques at the moment, or that we have had to date, really don't allow for a complex understanding of those cell interactions, the heterogeneity in these tissues. But by using these types of systems then and novel infrastructure, we get a better picture of the dynamics of the cells in their environment, which is obviously a really important factor in terms of model design. So for example, we could take, in the lab, obviously as Eoghan said, we work on testicular tissue, but we work on different urological tissues as well, either healthy or cancerous tissue. And what we can do is pull the cells out of these tissues and use techniques like single-cell sequencing to look at transcriptomic profile of these cells to give a greater understanding of the cell dynamics, but we can also take these tissue sections and we can use multiplex histology to actually get a better idea of the environment that the cells are in and how they interact with each other. So first on to the characterization. So this was led by Professor Paul Murray. He was really the main player in UL in terms of getting the funding for the infrastructure. And one of the main types of infrastructure that came in for spatial multiomics was the Akoya phenocycler fusion. And Paul Murray's lab has really been instrumental in developing this protocol here in UL in terms of allowing us to take tissue and be able to spatially profile it. And what this allows you to do is actually in comparison to standard immunofluorescence, where you have limitations in the number of fluorophores and targets that you can look at up to 100 plex proteins in the one tissue sample. And what this allows you to do is actually give you a full understanding of the proteins and the cells in their environment. So there's different systems that we can use here, and the main thing is that you get really nice, pretty pictures at the end of it. So even if you don't get something nice from the biology, you have a nice picture to put somewhere. And Dr. Aina Fennell really is one of the main users along with Ciara Leahy. They're both up there at the top in the lab, so if you have any questions, you could direct it to them. But what they've been working on is imaging these systems and then being able to analyze them. So what you can do is you can segment out the single cells from this imaged piece of tissue. You can then cluster these cell types, so you can look at different compartments of immune cells, structural cells. This allows you then to actually understand the cellular interactions because you know where they are in relation to each other. And this allows you to kind of develop an atlas of the tissue and understand the neighborhoods that the cells are in. And obviously this is very important because we want to know how does the environment influence the cell behavior? Because that's what we're trying to model at the end of it. So when we're trying to develop 3D systems, we need to know what are the important factors for driving the cell behavior. So you can understand the architecture of the tissue. So say, for example, this was in lung tissue, where you can see different features of the architecture that you can extract out from your multiplex imaging. We also have the capabilities then to look at cells at the single cell level using single cell sequencing. So we have due to arrive, not arrived yet, the Melody single cell FACS sorter, which allows you to pull out specific populations of cells. And we can use the BD Rhapsody single cell capture system, which attaches unique barcodes to each of the cells so you can identify them in your sequencing run. So this allows you to actually profile every cell in your tumour. And we can relate this then to spatial information by integrating the two systems. And then we can use an Illumina NextSeq sequencer that we have in UL, and this has different capacities, but ideally what we have here is an end-to-end workflow that allows us to isolate cells, do a single cell capture so that we can actually identify each cell in our tissue sample that we've taken. We prepare our sequencing libraries and run these, and we can do whole transcriptome sequencing or targeted sequencing panels to understand the profile of all of these cells. And obviously, as Eoghan already said, this is really, really important because when we're trying to think about our model systems, we need to understand where these cells have come from. We can't kind of go in blindly, and we're never going to really be able to recapitulate to the level that we want to, but at least if we're starting with these high-plex, highly informed characterization techniques, at least we can take aspects of that into our modelling. So one approach that we can use then is we can take native tissue. So if we're thinking about urological tissue, obviously where the cells are is really important. So if we take urological tissue and use these as our base tissue system to develop hydrogels or bioinks for printing parameters, what we can do in the lab is we can take native tissue, healthy or tumour tissues from different types of urological cancers or different conditions. So we can look at fertility in this instance as well. What we can do is decellularize the tissue, and we can take the matrix, which is basically what surrounds the cells, and we can take this and use this as our base material for our models that we're developing. And we can either load back in different cell types that we want in certain regions, and we can develop porous scaffolds, hydrogels, or we can develop them and try and modify them so that they can become a printable bioink for more highly complex models. So if we think about hydrogel approaches, if we just think of a very simple model where we're not really depositing cells in certain regions, but you can think of a bulk model system where we can take a hydrogel that's composed of different matrix components that are important to the tissue from which the cells are derived. We can put in urological cells, cancer cells, we can co-culture these with immune cells, whatever we've kind of found in our investigative approach in our characterization. And we can load these cells in and we can assess their behavior. So we just have some imaging of cells in these hydrogel systems, and you can look at the whole tissue there. So all the green cells are alive cells, which is obviously what we want. So, but if we think about more complex modelling, we can also use these ECM materials as bioinks. And this is really for the development of 3D printed models. So when you think of a hydrogel system, you're not really putting the cells in a particular area, you're just kind of bulking them in. You can kind of do inner and outer regions. But with bioprinting, it allows you to print very thin filaments that can be cell-laden. So you get very accurate deposition of bioinks, and you can develop complex models, and you can do layer by layer construct development, and these can be then developed at a larger scale. So you might not appreciate, but this was actually quite a good print. But for those of you that have ever tried printing, this was printing of a very, very soft hydrogel model using a fresh printing approach, which is you're printing into a free-form reversible bath. So it allows you to print very soft tissues, which was the difficulty in the first place when you're thinking of bioprinting. A lot of it was printing thermopolymers, and these are not very biocompatible. So we want to print very biologically soft tissues. But really the approach, I suppose, when we're thinking about bioprinting is what we need to do is to be able to define scaffold geometry. So we can import CAD designs to our printer. And what we would do is understand the tissue and understand what type of design we want. So if we know the matrix structure, we can incorporate that into a model design. We can also understand what type of matrix we want and what cells we want to load into this design. And then eventually what you would do is you would load these cells into the bioink. This can be extruded out of the print head. And what you'll end up with is cell-laden bioprinted constructs. So ideally what you could do is customize the matrix and the architecture, and you can see we get very high resolution, high print fidelity prints that have cells throughout the filaments. So you could layer by layer decide to deposit certain cell types in certain regions to understand those cell interactions, and then we can assess immunotherapeutics, chemotherapeutics, or different types of approaches there. So that's really kind of our approach that we've been taking, and that's kind of all from me. Thank you.
Thank you, Eoghan and Olwyn. So, questions for either of them?
Hi. So, after you do all this characterization and you get to the 3D printing phase, I'm wondering how big can you build models that represent the real organ or real part, and that can that be used to study the interaction of medical devices with those tissues and degradation and so on?
Yeah, I mean, that is the difficulty with bioprinting, is the scalability of it, especially with softer constructs. When you're thinking about thermopolymers, the scalability is a lot easier. You can incorporate bioinks into thermopolymer systems as well. But I mean, I guess it really depends what you're looking for. Are you looking to recapitulate an environment, or are you trying to scale up for regenerative medicine purposes? So a lot of the time with printed constructs that you want at a large scale, ideally they're something that you want to implant into a defect region rather than trying to develop an accurate model system. So if your angle is to develop a printed construct that will promote tissue regeneration, that's really where you'll be looking for the scalability. And what you can do is you can do full face scans of certain tissues. So you could take out, for example, say like intervertebral discs or meniscal tissue. You could take a full scan of the tissue, and then some of the printers that are available would be able to print the same dimensions and size as the tissue itself. I think ears, meniscus, and discs are probably the main ones that have been printed to scale, but not really with soft bioinks. So you're limited because the structure will collapse in terms of the integrity of the filaments. So I guess you're trying to weigh up. I'm not really answering your question, I'm sorry. You're trying to weigh up, I guess, compromising probably the architecture when you're trying to print a soft biological material. So in terms of scalability, on the soft side of things for materials, I'd say maybe, I don't know the size of them, centimeters cubed. I'd say like, maybe 10 centimeters cubed is probably, for the very soft printed constructs, that'd probably be the size. But you can print anatomical sized constructs, but probably not with very, very soft bioinks. But in the case of some of the disease models, we don't really need that. We can print very small structures to look at cell interactions, so we don't necessarily need to scale up on some of those, unless we were trying to incorporate maybe vascular systems, but we can develop channels within the bioprinted constructs, and then we can endothelialize them to actually kind of recapitulate a vessel within a construct. So I'd say you're trying to weigh up what you're trying to look at rather than the scalability of it, if that answers your question. Okay.
Yeah, I'd just add to that and say that, and say you probably wouldn't use this for evaluating medical devices. If someone handed us a medical device right now and said evaluate this, we would go straight to the abattoir and take the bits of the pig that they allow us to take and do the interactions there. This is very much to do with kind of, if you want to understand how a treatment or a very small device interacts with things on a kind of a cell or a cell neighborhood region. This is not to do with kind of a urinary Foley catheter or a pacemaker. This is if something was eluting out of that, then you would probably want something like this. It's definitely, there's an issue of scales, all right.
Could you grow cells to see what factor you're erosion or what pressure will cause that?
You could do that, all right, yeah. And I suppose that's the limit, say, of using an ex vivo model, is that you're not going to see the remodeling. Whereas in this you could take aspects of the device, like you're saying erosion or kind of seeing cellular injury, and you might take a very small portion. But yeah, you're as Olwyn said, you're limited on scale when you're doing something like this. When you're trying to, like the thing about modelling that's nice is that the more complex you make it, the better. Whereas when you're developing a treatment, you want it to be as elegant and as simple as possible.
Is that why you use the mechanical property?
It is indeed, yeah. You're looking at that. You want to look at the interaction. Yeah, you're at that level where the cell is interacting with its environment, and you want to see how a treatment interacts that, or interferes with that interaction. It's certainly not for kind of like the macro scale mechanics would be for looking at medical device interaction.
Would it be fair to say that, let's say, if we give you certain conditions of pressure and cycling that a device would apply to that tissue, you could replicate on that macro scale or small scale and say, well, if that happens in that small region, there's a chance that could happen or propagate.
Yeah, with some of the model systems, we could modify our culture parameters. So you could use a bioreactor system that maybe mimics fluid flow or cyclic loading, kind of while the cells are in culture, that might mimic the physiological environment. And the same with if you were doing, say, vascular systems, we could have perfusion models that mimic fluid flow with different materials that mimic the properties of different fluids. I would say from that perspective, anything.
You can certainly, in a model this complex, you can model certain aspects of a device, and that might save you time when you move it to an animal. But yeah, if you're looking at whole device interactions, you're going to need to have an entire tissue, if not an organ or an organ system.
Yeah.
Great. Any further questions? Cool. So thank you.
Thank you.
And so with that, I'd like to welcome Nico Walsh, who's going to talk history of urology and talent development, and also keep us on time.
Where am I going here? Ray, where do we go here? Lumped them all into one. Apologies, folks. Confusing everything. So yeah, now these slides, I could do an hour or 5 minutes on them, so I'm going to err towards the 5 minutes. And we had it down for 20, but we're kind of going a bit over and want to give people time for coffee. So excuse the expedited nature of this. And also, it's a little bit different. I kind of put it on at the end. The idea is that the guys were showing all the kind of capabilities and abilities that we have within the Bernal that could be used for urological research. This is then just one of my own projects that I've worked on for about 12 years. It's urology related, but it also focuses on the translational side of the stuff that I do. So my lab, there's two parts to it. One was translational research and the other was basic research. But the translational research in this project was particularly informed by 8 years and €1 million that I spent. It finished in 2010 on trying to commercialize a graft for arteriovenous fistulas. Or, well, a graft to replace the fistula. So it was a class 3 device, and at the end of it, showed that it worked a lot better than current technologies, but the amount of money needed to spin it out of the university was in the order of kind of $30 million, so it wasn't really that feasible. So we quickly stopped messing about with class 3 medical devices and moved towards a class 1 device. And so what it worked on is a safety valve for urology catheterization. And this was particularly then informed that, so at that time, there's a PhD student in our lab, Niall Davis, he's a consultant urologist now, he's going to speak to us after the break. But as part, or during his PhD, he was on call out in the hospital. Overnight, one night he got called by the urology team got called because somebody inflated a balloon of urology catheter in the patient's urethra. So he came back out to us the next day, grabbed a bunch of engineers, surely we can come up with a solution, which we did. And so then we've spent the last 12 years trying to get this commercialized. So again, so just in case people don't know what urology catheters are, there's effectively these long tubes that go in through the water passages, an anchoring balloon at the end of it, and ideally the anchoring balloon will be inflated when the balloon is correctly positioned in the urethra to stop it being pulled out. And if it's in the wrong place, lots of things can happen, all the way up to urethral rupture, depending on whether the patient's awake or asleep when it's done. And so our solution is a very simple device, but it's not so simple. But, and I'll explain to you why that is. But this is effectively it. It's a safety valve that sits between the syringe and the catheter. There's nothing complicated about it. So when we came up with our first idea, it was 2011. So this is me. See, I didn't have any grey hair back then. This has upset me hugely. And that's Niall. And so this was also out in the Cleveland Clinic. So they sponsored that award. And so yeah, we didn't have, there's two parts to the device. One is the safety, is the kind of the threshold pressure that it activates at, but also the key point of it is that what we figured out after a while is that when we did the initial search, what won this prize, we found a 1978 patent that it had the exact same idea. And kind of thinking, well, if 40 years, 50 years ago, they came up with a solution for this problem, why is it still a problem? And so what we figured out, of course, is that the pressure required to inflate these balloons is a function of the user. And that was the key thing that we kind of looked on. So these are 8 PhD students, 4 male, 4 female. These are the 4 men on top who kind of show off and how strong they can depress the syringe or plunge. And the 4 female PhD students are much more collected and comfortable with themselves and just take their time. And the interesting thing about this, right, is that if you just set a threshold pressure, where would you set it? Because if you set it up here, then that means you could cause a lot of damage before it activates. Whereas if you set it down here, each one of these would cause a false positive, because if whoever presses the syringe really hard would activate the valve. And so what we ended up doing, and this was the key piece for our invention, was putting this flow restrictor before and to stop any heavy-handed users creating pressures that would falsely activate the valve. So that was really what we did. When we did a load of different things in the lab, or within what we could do as engineers, so we did some cadaveric testing. Excuse me. And we tested some stuff in the lab. So this is testing in porcine urethras with a 1 mil inflation, and then if you go to 6 mils, you can see some extravasation there of the dye, and then you can kind of see complete rupture as well here with the 10 mils. So that was the work that we did with porcine work. So when you look at that as on a graph, this is kind of what you come up with. So the idea here is these are the ones that didn't rupture are ruptured, and these are the ones that didn't. And so we were kind of getting towards a number that the threshold should be at. And so we tested a load of catheters. These are commercially available catheters, and they were all kind of at 120 kilopascals. Then we noticed that these ruptures, noticed in the porcine urethras were over 150, and so we set our threshold pressure to that. And so then we developed the device, and then we started looking at testing it. This is testing it in human urethras that were taken from transgender patients undergoing surgery in Germany. So this is a collaborator of Niall Davis's. And so we tested that work in the lab as well to show how different types of damage you could get. And then this science stuff over the last few years, it's kind of been published and won some awards, and it was kind of doing it, still it's 5-6 years ago. So we got our patents granted in Europe, US, China, and then it comes down to economics. So medical device design, it took me that 8-year, €1 million traumatic story to figure out medical device design. Very little to do with design, and mostly it's to do with economics, particularly for spin-outs. And so the cost benefit of the solution. So we monitored 2 hospitals in Dublin for 6 months at injuries, and there was 37 injuries divided by the number of catheterizations, gave us actually the extra cost on top of the catheterization kit was almost 10-fold increase in what the patient cost to catheterize everybody if you take those injuries and add it to the cost. Now, the interesting thing is we thought by coming up with a device to save these costs would be a good thing, but turns out the hospital don't really see those savings because somebody else just goes into that bed anyway. But it means you get more patients saved, but these are nuances in these arguments. But in our prospective study, we measured the kind of what happened with the patients, and then we followed up these patients in 3 years later, in 2019. 2 things were surprising. One was basically 80% of the patients had new onset stricture. So you always wonder why stricture disease happens or is caused, and may well be through interventions that lead to strictures being caused. And then just 1 patient actually died due to urosepsis following catheterization. So it was kind of a very sobering findings from that. And so then we did another prospective study. We found 66 injuries across 6 months in 7 institutions. And we did a bunch of follow-up, but really take-home message, right? So these were some institutions in Ireland, there were some in the UK, and we kind of developed this metric of number of injuries per month per 500 beds, because all hospitals are different sizes and it's hard to compare one to the other. And so if you use that, this study was an average of 3.1 injuries per month per 500 beds, and our 2016 study was 3.2, which was kind of interesting that data kind of repeated itself, because a lot of people deny these injuries happen. And so we did a trial then in the last year, or well, it's 2 years ago now. So we had 6 institutions in 12-month study, and primary endpoints was urethral injury, and then we wanted to also know if there was ventings. And our trial showed that there was 1,000 catheterizations, and there was 24 ventings. So that's an incidence rate of about 2.2%. Now, there was 3,000 valves used in that study. So we're just assuming people used them, they were uneventful, and so then they didn't return the data. So really the injury rate is probably 0.8 to 2.2%. But the interesting important bit for us is there was no injuries in the 24 ventings. And so this is just an image of a patient with the bladder. This is the catheter, and this is why you can see it happening. So the balloon is inflated here, because the tip of the catheter could be in the bladder and draining urine while the balloon is still not in the bladder itself. And so the 150 kilopascal, so we did all our science that we did here that I showed you earlier on. Then we've got the device itself. And then finally, because we had 24 ventings and 22 of the 24 of them, there was no pain, we argue that this validates that the threshold is a good threshold because there was no damage to patients. The reason I'm labouring this point is the FDA is arguing with us at the moment. So it doesn't have FDA approval, but it has CE marked nearly 2 years ago now. 12 months ago now, but only because how do you objectively verify that there's no damage caused when the balloon inflates? And that's very difficult to do, because what are you supposed to do? Catheterize the patient, the balloon inflates in the wrong place, then take the catheter out, put a camera in to check there's no damage, and then recatheterize. So it's not really feasible, certainly for us to try and do that. But the other bit I'd labour the point on is just health economics is going to be a big thing in all medical devices going forward, and there's quite robust models on that. We used the York Health Economics Consortium, and they effectively showed just over a 1-year cycle, this is the cost of our device, which is much higher than theirs, but the overall cost of using a safety valve saves effectively 50% of the cost of the treatment. So we got our certifications, and so we were the first, our spin-out company is the first company in Ireland to be approved for CE mark on the new medical device regulations in Europe. And so we're still fighting the good fight with the FDA at the moment. But effectively, that's what our device looks like. So it's a catheter kit that has a safety valve included in it. And so I suppose the point with what I wanted to show here is that UL, we have a rich history of doing applied research and supporting industrial funded research, but also quite a strong track record in kind of supporting spin-outs and any kind of, if there's developments from technologies and grants and things like that, that's another mechanism that we can use. And so this was us in 2011. We went back there 8 years later. So the grey hair firmly sitting after that. And Niall is there just on the right-hand side as well. And so, yeah, it takes a toll, all this stuff, folks. So that's it from me. Cheers.
Great, thank you, Mika. Any questions? Mika. I'll just give one anecdote. Mika was my FYP supervisor, and I did have hair at that time in 2007. So
where did it all go wrong, Sean? The two of us.
Any questions?
Done.
Oh, Sarah.
Thanks, Mika. How do you make the decision to go from just doing applied research to moving it towards that level of actually getting approval for a medical device?
How do you make the decision? I thought lots of people don't. I mean, it's very PI specific as to how they see. A lot of people do basic research. I mean, there's quite a few PIs. It's interesting if you ask them, think that if we develop IP in the university, it should be just given away because it's state funded, etc. The university have a different opinion on that, though. So they like to maintain and support their IP and try and commercialize it to get money in. So it's very PI specific. I mean, I was naturally drawn towards it. The device that I talked about that upset me after 8 years, that came out of my PhD, was a solution to a problem that I was looking at, which was bypass surgery. So I suppose it's just the idea of, everyone talks about the impact and it's all translational research and all that sort of stuff, but they're getting something. So our device has been used on 15,000 patients in the last 12 months. So even just on the very low injury rates, there's probably 150 people walking around whose urethras aren't injured because they use our device. So it's a kind of a nice thing to think about. I mean, it's kind of part of it is perspective and age as well. When you're all like me, I mean, I've graduated a bunch of PhD students. My H index is fine, so I don't care if it goes from 40 to 50, it makes no odds to me. So now the idea of having that side of an impact is interesting.
Great, thank you.
Paul Burke, Chief Academic Officer for the Hospital Group.
Burke, how are you?
I'm well, Mika.
Good.
The obvious answer to your question to Mika was the clinicians will tell you if the thing is going to run or not. I'm very familiar with Mika's first case, which was really part of the hospital and the researchers starting to get together for the first time, and Mika was one of the standard bearers of that. But that AV fistula model was never going to run from a clinical application point of view. Yeah. This is very straightforward. It's a simple concept for a common problem. And the clinicians are the people who tell you this will run. So the key is good researchers with good clinicians who have an interest in research, who identify clinical application, and that's the formula, in my opinion.
No, absolutely. I remember, and it is that. It's but I also think I would be of the considered opinion that Class 3 medical devices don't really, I suppose, be commercialized into startups. It's really the realm of the proper grown-up Boston Scientifics of the world who have the resources to put behind something that they believe in. So, but yeah, absolutely on both.
Just a follow-up, Nico.
Sure.
This is mainly applicable in male patients. It isn't as big an issue in female patients, presumably.
Yeah, no, completely. So even the trial numbers that we use, we're kind of being a bit bold with it because we just divided by, because all the 37 injuries in 2016 were all male patients. It's it's obviously, but it's just an anatomy thing. It's I think the idea, there is female catheters which are much shorter, but I think they might be being done away because sometimes people can take those and incorrectly use them in male patients. But this is 90% and an older male problem as well.
Great. So thank you very much, Mika.
Cheers.
So I think that concludes this session. So I want to say thanks a million to all the speakers. It was fantastic to see the collaborations that we have ongoing, and I suppose the diversity of the research and the quality of the research that's happening. And within that, as I said, please come talk to us if you have an idea, if you want to come work with us, if you want to come be part of our collaborations. We're always willing, and we'll be outside for the further tea and coffee. So
It's kind of 25, just a bit conscious of time. So there's tea and coffee, I think it's outside the second floor, which is down the stairs and out the sides, and we're going to kind of come back in about between 5 and 10 minutes, so you can maybe bring it back in with you. All right, folks, thank you.
Will I go? Great. Thanks, Ray. I've got the nod from Ray to get started. So good afternoon, everybody. You're welcome back to the second part of this overall session on urological research. And after the focus we've just had on current urology research at UL, we're now going to look at the future directions of urology research. And the format of this afternoon's session is that we're going to have two keynotes from Mr. Niall Davis, who's a surgeon at Beaumont Hospital. And then we're also going to follow that with another keynote from Robert Lund, who's the director at Boston Scientific. And I'll say a little bit more about both of them in a moment. And then we're going to move to a panel session, which is going to feature Ivania and Marina, who's a urology research and development engineer at Boston Scientific, Jason Kenny, who's joining us from Bon Secours Hospital Group, and then also Niall and Robert will join the session along with Nico Walsh, who you have heard from earlier this afternoon. So I'm delighted again to chair this session. For people who don't know me, my name is Norley Kennedy. I'm the Vice President for Research at the University of Limerick, and this is one of our feature events as part of our Research Week, which across this week is featuring research from across the university and I suppose showcasing the excellent research that the university is currently undertaking with a range of partners, many of whom are featured here today. And I think that's a key part of this afternoon session, is to look at industry collaboration, to look at how clinical, industry and academic partnerships develop, how innovation comes from that, and how they've led to some very, very successful outcomes, as you've seen earlier from Mika's work. So an opportunity to discuss a little bit more of that this afternoon. So on that, I would like to welcome Niall Davis. So Niall is a graduate of University College Dublin, and who undertook his basic surgical training with the Royal College of Surgeons in Ireland and completed his higher surgical training in urology in 2017 and completed his fellowship in minimally urological surgery in Melbourne in 2019. Niall is also a senior lecturer for the RCSI and works as a consultant urologist in Beaumont and Connolly hospitals. And he's completed a PhD in tissue engineering in UL and has over 150 publications in index medical journals to his name. So we're delighted to have Niall with us. And Niall, on that, I'll invite you down and you can get started. I think everything's ready to go for you. And then we can take a few questions just after immediately your session if you need, and then we can move on to the panel session after that. So with that, over to you, Niall.
Thanks. Thank you. Hello, everyone. Thanks for inviting me to your Research Week. It's been a long time since I've presented here, so I'm delighted to be down. I see a few familiar faces in the audience. So just over the next 20 minutes or so, I'm going to talk about what I do as a surgeon day to day and how we try to collaborate with industry and universities for research purposes. So the title of my talk is Future Directions in Urology, Research, a Clinical Perspective. So just where I think my specialty is going to go over the coming years. So over the next 20 minutes, I'll give you all an introduction to urology. I'll briefly talk about some recent technological advances in urology, some in vitro and in vivo collaborative research that we're doing at the moment in conjunction with UL and with Boston Scientific, and then some future projects that are in the pipeline. So I'm Niall Davis. I work as a urologist in Beaumont Hospital, and I have a heavy research affiliation with the University of Limerick and with the Royal College of Surgeons in Ireland. So on a day-to-day basis, I work as a surgeon, and to become a surgeon in Ireland, you typically do about 5 or 6 years of basic surgical training and then travel abroad where you tend to subspecialise. So I went to Australia, specifically I went to Melbourne, and the hospital that I did my subspecialty or fellowship in was called the Austin, and this was in minimally invasive approaches to common urological problems. So as I mentioned, I'm a urologist, and this means that I specialise in the urinary tract. So any disorder, benign or malignant, involving the kidneys, the ureters, bladder, prostate and urethra typically comes my direction. In layman's terms, I'm basically a plumber. So fix the human plumbing when it goes wrong. As a specialty, we subspecialise. So there are 7 different areas in urology, and the 2 red ones are the areas that I've subspecialised in. So uro-oncology, which is cancer, and endourology or urolithiasis, which is kidney stones. More recently, urology as a surgical subspecialty has really embraced technology. So we do a lot of robotic surgery or keyhole surgery, which would be known as minimally invasive surgery. And as a subspecialty, we tend to be heavily involved with innovation and academia. So there are 2 other parts to my specialty that I partake in regularly. So what does uro-oncology and robotics mean, and why was I drawn to it? So previously, if someone had a kidney cancer or a disorder with their kidney that necessitated an operation, they were typically left with a big open scar that took quite a long time to heal from and was aesthetically displeasing. So as surgery has evolved, so has approaches to kidney disorders or kidney tumours. And one area is robotic surgery. So this is the Da Vinci robot that we use in robotic surgery. It's a minimally invasive approach for treating kidney cancers and upper tract urological disorders. The advantages with the robot are that patients typically recover quicker from their surgery, they're out of work for a shorter amount of time, and it leaves basically no scars or very small scars. So there's a Da Vinci robot in a lot of hospitals in Ireland. We use one in Beaumont, and probably the one that's in UHL has been established for the longest period of time in Ireland. So this is a picture of the surgeon typically at our console. So we're doing the operation. It's a bit like a computer game, and you can see the robot in the background, and we control the arms of the robot with this console, and we're able to see into the patient's body through this console here, and there's a camera attached at the patient's bedside. So for most of my operations in Beaumont, I'm usually sitting at a console operating it, and the robot's doing most of the work. So that's where I really see upper urinary tract surgery going. It's becoming more and more minimally invasive as time progresses. It's very well broadcasted in the media, and patients in Ireland, when they have a kidney issue that necessitates a surgical operation, a lot of them are looking for robotic surgery at the moment, so it's nice to be able to offer that to them. It's the same with kidney stone surgery. Up until about 20 years ago, if you had a big kidney stone that was blocking or obstructing your kidney and it needed to be removed, again, it necessitated a big open incision across your flank. With kidney stone surgery, it's become even less invasive than the robot. So now if you have a kidney stone, due to advances in technology, we're able to pass the camera all the way through the water passage, all the way up to the tube to where the stone is. We can sometimes put a basket around it and take it out. The stone is too big to come out. We have a little laser fibre that can go through the camera and laser the stone into dust, and the dust drains by itself. I had a picture there of a big kidney scar. Now what we're doing for big kidney stones that are up in the kidney is passing or drilling a hole into the kidney, passing a camera through it, and then lasering the stone and extracting it through the flank. The patient's left usually with a scar that measures about 1 centimetre. So there's some of the nice aspects of my day-to-day job. It's very minimally invasive. Patients recover quickly, and the technology is getting more intricate, and it's very enjoyable to work with it. So as I mentioned at the beginning, I also have an interest in academia and innovation, and about 25% of my working week is with academia and innovation, usually with the Royal College of Surgeons or here in the University of Limerick. So it's not possible to do proper academia or innovation by yourself, to do good research, to publish high quality papers and to do good clinical trials. You need to have a good team around you. So typically involves a scientist, a biomedical engineer. I don't tell the biomedical engineer how to do their work, and they don't tell me how to do my operation, because I know that they're better at what they do than I am, and they probably wouldn't be able to do the operation if they tried it. Anyway, so I work predominantly with Professor Walsh. I've done a lot of work with Eoghan Cunnane, who's there, the Tissue Engineering Research Group, which is Fergal O'Brien's group in the RCSI, and the Bernal Institute here in the University of Limerick. We have regular meetings and come up with ideas and projects to try and advance or develop urological surgery bit by bit. So at the moment, we're doing a lot of interesting in vitro and in vivo research. We have kind of a protocol or a pathway up in Beaumont now, where some of our trainees are taking time out of their training to spend a year or two learning research techniques and trying to really develop their sub-interest in urology. A very important component to good research is to set up a biobank, so to try and get proper animal or human tissue that you can do your research on, because it really mimics clinical settings and in vivo or operative settings. So this is a picture of an explanted urinary tract from a pig, and we've set up a pig biobank down here in Limerick with Olwyn and John Mulvihill. You can see the two kidneys here. This is looking through from the back. That's why the left kidney looks like it's been mislabeled, but it hasn't. So these are the two kidneys and the tubes that drain the kidney. These two tubes are called the ureter, and the ureters are typically where kidney stones get stuck when patients are passing them. As I mentioned at the beginning, when we are treating kidney stones, we tend to laser the stone. But one interesting thing is that the laser doesn't always hit the stone. You can't hit the stone 100% of the time. So sometimes when the stone is in the ureter, we actually hit the ureter itself, and we're not sure what kind of damage this does to the ureter. So at the moment, we have taken human ureters. So when I take out a kidney for a kidney tumour, we have patients who consent to donate their ureter with the specimen and allow us to perform research on these ureters. So we're intentionally lasering the ureters in the laboratory to see what kind of effects that the laser has on the ureter, and then trying to develop a method for preventing injury to the ureter. In addition, when we do kidney stone or robotic stone surgery, the instruments that we use, if you can imagine, we're passing rigid cameras and semi-rigid cameras up this very fine tube. The human ureter is only about 4 millimeters, and we exert a lot of force on the ureter. Occasionally we injure it, or we even avulse or disrupt it from the kidney, which is disastrous, but it does happen in Ireland every year. So we're actually testing the mechanical properties of the human ureter as well to see what type of force it can withstand before it becomes injured or avulsed from the kidney. The ultimate aim then is to try and have a mechanism on an instrument that alerts the user that they're putting too much force on the ureter when they're operating. So involved in this project is Professor Walsh, Eoghan, Cunnane and John Mulvihill, and one of my trainees who's taken 2 years out to investigate this in more detail. It's again as part of the biobank. When we do kidney stone surgery, we typically leave in a temporary stent in the ureter at the end of the procedure. This stent goes all the way up to the kidney and it keeps the ureter open, and this allows stone fragments to drain. With the stent, we're not sure what kind of effects it has on the ureter when it's in. Does it inhibit peristalsis of urine? Does the patient's body try and reject it? So that's another project that we have ongoing here in the University of Limerick. Most of these stents are from Boston Scientific and various companies, so we're able to see what different types of stents and how they affect the urinary tract. So that's just briefly some of the in vitro research or benchtop research that is taking place at the moment. We have another trainee who has taken time out to do a PhD specifically on urological research, and she's heavily involved here in the Bernal Institute and also in the RCSI. And her project is in vivo. So her project is very interesting, and it's really come along a long way. When we do kidney stone surgery, to see the stone, we need good vision. So we typically have an irrigation channel going through the ureteroscope up the ureter to the kidney, and the irrigation is constantly going and it's constantly flushing through the urinary tract. As you can imagine, if you have constant irrigation in a closed system, pressures that the kidney is exposed to could potentially be quite high. So we don't know what type of pressures that the kidney is exposed to when we're doing kidney stone surgery. We don't know if high pressures are associated with patient injuries or morbidities, or do high pressures cause patients to have infections afterwards. So in Steph's project, we obtained a research grant from Boston Scientific, whereby they gave us these are coronary artery pressure transducers. So these are pressure wires that go into the coronary arteries, and they typically measure cardiac pressures during angiograms or angioplasties, so during cardiac surgery. But what we've been doing is passing these pressure wires all the way up the ureter at the beginning of our kidney stone surgery, and we're monitoring what type of intraoperative pressures that the kidney is exposed to when we're operating on kidney stones. So here you can see the coronary artery pressure transducer has been passed all the way up into the kidney, and we leave it there for the duration of the procedure, carry out the procedure as normal, and then look at our pressure tracings. We then follow up the patients afterwards to see, are the patients who are exposed to high pressures, do they experience more adverse outcomes? So this is a video where the surgeon has been blinded. They're operating away as normal. Before we started this in vivo project, animal studies or anecdotal studies suggested that the kidney would be exposed to about 20 to 40 millimeters of mercury during a kidney stone operation. But when we're using our wire, you can see that the pressure is going up and up and up and up and up. So we're hitting about 200 millimeters of mercury, which is extremely high for some of our kidney stone surgeries. And this is something that has never been investigated before. So we have used these pressure wires from Boston Scientific in about 150 patients now. And these are some of our results. As you can see, the patients who are exposed to higher intrarenal pressures, they're more likely to develop a pyrexia or a temperature or become septic and be readmitted to the hospital. And I guess the ultimate goal for this project is to have, again, like our benchtop data, a little sign or alarm on the instrument to say to the surgeon, you need to maybe turn down the irrigation a little bit, because if the pressure that you're at continues, the patient is highly likely to experience an adverse outcome and possibly become septic. These results are actually being presented today in Chicago at our AUA, American Urological Association meeting, and there's a lot of interest in them, which is great to see. It's great to have good Irish research being presented internationally. So there some of the benchtop and proper human in vivo studies that we're doing at the moment. We have another number of side projects and future urological research projects that have started to take place, or their wheels are in motion. Our next project is it's one that I'm very fond of because it mixes both of my subspecialty interests. It's a mixture of robotics and kidney stones. So I've spoken a lot about kidney stones and kidney stone surgery, and sometimes if a patient drops a kidney stone and they don't get rid of it and it becomes stuck in the ureter, as a consequence to it becoming lodged or impacted, the native tissue doesn't like this and it shrivels up afterwards, or it becomes ischemic. As I mentioned, the ureter is only 4 millimeters in diameter, so it doesn't take a lot to compromise it. The incidence of ischemia after kidney stone surgery it's as high as 3%. At the moment, the gold standard surgical procedure to fix this ischemic segment is to incise it or cut it out, and then to get a patch of healthy tissue and stitch it over. And this is very difficult to do because the ureter it's tiny. It's 2 to 3 millimeters of tissue that you have to work with, and you need to have very good vision to be able to do this operation. And that's why it's been done robotically at the moment, because the camera that's on the console can go right up to where the injured segment is, and it gives the surgeon good vision on where to put the patch and how to stitch it in. One disadvantage with this procedure is that this patch, it actually comes from the patient's own mouth. It comes from buccal mucosa, because the mucosa of the mouth is able to accommodate contact with urine. It responds well to it. But disadvantages with the patient's own mouth is that, as you can imagine, you only have a very limited amount of tissue to work with, and there's a high chance of causing an injury to the patient themselves when you're harvesting the graft. There's a lot of salivary ducts and salivary glands in this area, and they can occasionally become injured during the harvesting process. So we've launched a grant proposal here in Limerick in conjunction with the RCSI to try and develop a biomaterial instead of using the patient's mouth, that you could perhaps develop in a laboratory, take it off the shelf during the procedure and implant it onto the diseased segment instead of using the patient's own buccal mucosa. So this is the theory behind the idea. The surgeon's at the console, The robot is here, patient is here. We take our graft from the shelf, put it in, and then stitch it onto the diseased ureteric segment. So there's some of the research ideas that we have that are ongoing, some of the benchtop, some of the human, and some of the future research projects that are taking place. I mentioned at the beginning, I think that the future of my surgery, or my surgical subspecialty, is becoming less and less invasive, and patients seemed to be on board with this, because it's amazing the amount of people who have read up on their own urological conditions and seek a more minimally invasive approach to have the problem fixed. When we're doing these research projects, I think it's very important to publish them and to have them peer-reviewed as you go along, because they add a lot of merit and credibility to what you're doing. Because if your projects have been reviewed by a journal or an editor from a different country, it really validates that you're heading along the right pathway and that your research is good and progressing correctly. These are just a snapshot of some of the publications, recent publications. That's one with myself and John Mulvihill. Most of the others are with Michael Walsh, Owen. I can see Cunnane on this one. Don't know where Owen is. I'm sure he's on it somewhere. Anyway, so that just concludes my presentation. I just wanted to provide you all with an introduction to my specialty, what we do, how we do it, and what we do on a day-to-day basis. Just to emphasize that surgery in general is becoming more and more minimally invasive, and it's important that the universities realize this, because this is the way that surgical research and collaborations with universities and industries should be tailored. I've spoken about some of our in vitro and in vivo collaborative research projects that we have ongoing with Beaumont, the University of Limerick, and RCSI, and this network seems to be working very well. It's great to have opinions from different people in Ireland who are key opinion leaders in their field, whether it be surgery, biomedical engineering, science, and then I concluded with an example of a future project that we hope will take legs pretty soon, and maybe in a year or two I'll come back and present our findings from that one. Thanks all for staying on.
Thanks very much, Niall. I'm delighted you chose Limerick over Chicago.
Oh, yeah.
Great, thank you. You can have a seat, and we'll sit back down here in a few minutes for our fireside chat. Thank you very much, Niall. It's just fascinating. It's wonderful to see the developments over for urology surgery, and particularly robotics, and the importance of, I suppose, the patient at the centre of those decision making to ensure their quality of life and their experience post surgery is considered. So thank you very much for that overview. I'll move on now to Robert Lund, and Robert, to introduce him, is the Director of Active Implantable Development for Urology at Boston Scientific, and he's based at their site in Minnetonka, have I said that right? Minnesota, and you've been over 26 years in Boston Scientific with expertise in their men's and women's pelvic health, medical device development and regenerative medicine. And Rob, I'm advised that you're also a very excellent producer of wine in your own right, and Ray has advised that we talk to you specifically about that as well afterwards. It's a pleasure to have you here, Rob, and come forward and you can get started with your presentation, and we'll move on then to the far side. Is it obvious what I do here to move on to the next one? Yeah, straightforward. There you go. Perfect, thank you.
Excellent,
okay,
great.
Rob?
Yep, sounds good, perfect. Well, thank you everyone for having me here today. I was telling Sean earlier today, I said, I think I'm finally adjusted to time here in Europe, so I know it's been a great time in Israel, or here in Ireland, and a great time here at Limerick. So what I wanted to talk today a bit about, whoops, going the wrong way, is talking about erectile dysfunction. And this is probably one of those disease states that if you talk about 25, 30 years ago, people never even talked about it. You did hardly talk about it with your spouse. You would certainly not talk about it with your family or your friends because there was a real stigma around it. But as you see from the data here, that really 1 in 5 Americans, men, in the data that we have here, have some form of erectile dysfunction, and more than half of men over the age of 40 experience it to some degree as they go forward. And really the causes of that are anything from heart disease to diabetes, cardiovascular disease, and folks with spinal cord injury, which causes it. And what I want to do is take you a little bit through the patient journey, because I think if you understand the patient journey, you understand how profound of an effect it has on them. So what I want to do is take the example of a person who was diagnosed with prostate cancer, right? So they had a prostate cancer, they probably had a Gleason score of 7 or higher, and their surgeon recommended that they get their prostate removed. And at that particular time, that patient's thinking, I want to save my life. I need this surgery to save my life, right? And they go and get their prostate removed, and as good as the surgery is, and as good as the robot is, there's still a lot of men who have nerve damage and are either left with incontinence, erectile dysfunction, or both. And so for these patients, now that their lives have been spared, they're left less than whole, right? So they now say, what do I do with the ramifications of leftover of my surgery, right? So the first place that urologists and their primary care physicians start is prescribing a pill, a PD-5 inhibitor, Viagra, Cialis, Levitra. Those are the drugs of choice that they try. And for people with a mild to moderate erectile dysfunction, it works really well. They're great pills, they have the effect. But still, every time that you want to have an erection, and we got to get comfortable with saying the word erection in a presentation, so you'll maybe hear it a few times along the way, they got to take a pill 30 minutes to an hour ahead of time, right? Then you have the folks that, in the case of this radical prostatectomy where they had nerve damage, they're probably having severe erectile dysfunction, and essentially it's not going to work for them, right? So then they got to go to the next line of treatment, which is either getting an injection where they take a needle and inject a drug into their penis every time they want to get an erection, or a vacuum device where they force tumescence through a vacuum so that they can get an erection, or they'll go with some sort of urethral suppository, which will also generate an erectile response. It's not a great solution for people that they want to do that. So a lot of people continue on with their treatment journey and they find a penile prosthesis, okay? And just to kind of put in your mind around this, from that time the person had their radical prostatectomy to the time that they probably got to a penile prosthesis, the average amount of time is 6 to 7 years. So imagine the effect that's had on the individual feeling less than whole. Imagine the effect it's had on them and their partner. And when they get in a penile prosthesis, which I'll go over here shortly, their lives are changed and they're brought back to whole, right? So if you kind of say, well, geez, how big is this problem? I've got U.S. data here. Basically, starting with men over the age of 18, which is 124 million in the U.S. And if you talk about people with health insurance that are going to be going down the treatment pathway, it's about 112 million. When folks that start seeing their doctor about ED, it's about another 10 million. And men that have been diagnosed with severe erectile dysfunction is about 1.9 million. And men who are actually seeking, what can you do for me, is about 1.7 million men in the US. And when you look at the penile prosthesis, we only implant, as an industry in prosthetic urology, about 30,000 units a year, about 2% of the population. So when you say, well, only 2% of the population is getting this, what can we do to improve that, right? And we're going to talk about that today as well. So let's talk about the inflatable penile prosthesis. This was actually invented 50 years ago. One of the things that's going to be going on at AUA in Chicago is we're celebrating 50 years of the penile prosthesis invented by F. Brantley Scott in the very early 1970s. And basically how the device works is there's basically 3 components to it. We have a reservoir, which gets filled with saline. which is attached to a pump that gets implanted into the scrotum, and it gets attached to 2 cylinders that go into the corpora cavernosum, which is in the corporal bodies of the penis. And how it works for a patient is a patient, when they want to get an erection or have tumescence in the penis, they'll take that scrotum, or they'll take the pump that's implanted in the scrotum, and they'll start squeezing it. And every squeeze, they're moving more fluid from the reservoir into the cylinders. The cylinders expand in girth, creating that tumescence. And for patients who get this device, they are highly, highly, highly satisfied because it works 100% of the time. We have patients come into Boston Scientific and tell us, wow, this is, you guys have changed my life, right? So you might say, Rob, okay, you have a device that's been around for 50 years. With patients are highly satisfied, how do we make it better? And with everything, you'll find that when you partner with industry, and you've heard it in previous talks, we're trying to identify what's that unmet need that either can make the outcome better or improve the quality of life for the patient. In the case of the penile prosthesis, the most difficult part is them manipulating the pump in their scrotum. So imagine that pump. You have a man, let's pretend they're a US man that maybe has a bit of obesity to them, right? They're an older man who's starting to lose some of their dexterity in their fingers. They have to find their pump in their scrotum, stabilize it with one hand, and squeeze the pump without being able to see what to do. And if you say, well, how spontaneous is that? How difficult is that? The answer is it's not very spontaneous, and for a lot of men it is difficult, even though once they get a hang of it, they're very satisfied with it, right? So when you talk to physicians, what do they not like? They don't like dissecting into the scrotum. It's a really sensitive area of the body. Patient training. Lots of these patients come back 3-4 times into their office, sometimes as high as 6-7 times, to get retrained on how to use their device. With patients, the manual dexterity, if you don't have it, you could be contraindicated for this device. Concealment, even though it's inside the body, I would say it's not very natural to reach your scrotum and start squeezing, because these devices take anywhere from a minute to a minute and a half to 2 minutes, depending on the size of the cylinders, to inflate. So the Boston Scientific solution that we're actively working on here in Clonmel, Ireland, and doing research on here at the Bernal Institute is on a electronic inflatable penile prosthesis. And what our goal is to get that pump out of the scrotum and replace it with what looks like a pacemaker and control it with your smartphone. So now when a patient wants to get an erection, they'll open up their smartphone app, they'll hit the button, they'll close their phone, set it on their nightstand, and get back to the moment that they were in. And that device will then, there'll be some automatic pumps, which we'll talk into here a little bit more, that will go ahead and do that same thing that the pump did in the scrotum, but inflating it automatically as they go forward. As a result of this, it takes energy to do that, right? So when you use your finger, you're the one providing the energy, right? So we're gonna have a implantable battery in that can, and it's gonna be rechargeable. So after a certain amount of uses, the patient will take this one that you see at the bottom, the black chunk of plastic, and they'll hold it over their device. And in about 30 to 40 minutes, they'll recharge their device and have a full new set of battery for doing it. And you say, well, okay, so that really meant a lot of unmet need, but one nice thing with invention is that it opens the doors to what's next after that, right? And so that kind of gets us to the improved outcomes that we're going to see below, because we're going to know what's going on. And really, the key as you get to the 9 o'clock position on this chart is that we now no longer have to do a programming for one size fits all, but we can get it down to individualized medicine. Patients are ecstatic. Even if it's taking them a little bit longer to inflate, they're not concerned about inflation time. What they're concerned was, how long did they interact with the phone? So for them, they're interacting with the phone for about 15 seconds. So they actually view the device as inflating faster, even though it's going to take slightly longer than what they were doing when they were manually pumping it. So it's incredible technology around piezos. There's a ton of research, as you've seen, that's going on here around it. but you kind of see a cross-section of how it's going. So I think that was my last slide. And I just wanted to say thank you for letting me present here today, and it's been fantastic to be here. So thank you.
Great, thank you very much, Rob. Fascinating talk, particularly emphasising the unmet clinical patient need, the importance of the patient, of the heart, user design, involvement of patient across the journey, but also the interdisciplinary approach to solving really important problems for people, often that are very silent and, as you say, often quite taboo. So wonderful, thank you for that. I think Ray wants to, at this point, just before, when I get the panel set up, and invite our panel members and introduce them. Ray is just going to get set up to show a video. As many of you will be aware, we have a new hospital being currently built over at Northern Trust on the Grudy Road, and we're delighted to have the CEO of the Bonds Secure Hospital Group with us here today, Jason Kenny. So we think it's just valuable, given what's happening on our doorstep, that we could showcase some of that. And as we get that going, yeah, if you do that now, actually, it might be the handiest. Perfect. Great. Just give you an oversight of what's currently happening behind
part of our
barriers.
2025 plan, 190 million investment in what will be a major development for the region to provide good help to those in need, to work with our colleagues in the public system and to meet the challenging needs of the population here in Limerick.
This state-of-the-art hospital facility really sets a new bar in terms of hospital design. In terms of sustainability, the credentials of this hospital really are above anything delivered to date in this country.
This building is going to change not only the face of Limerick, it's going to change the services and the care that will be provided for the people of the Midwestern region by the Bon Secours hospital system.
Well, it provides fundamental change for the delivery of healthcare in the Midwest. For our patients who have private health insurance or seeking private care, it allows them access to a whole range of medical and surgical specialties here locally in Limerick. It will prevent them the need to travel further afield to other cities in Ireland and beyond to get their health care, and they'll be able to do it in a timely fashion.
We look forward to providing new services, new cardiology, medicine, and things we haven't provided before. There's great opportunity for Limerick and for healthcare in Limerick.
Limerick is 30 years trying to get a private hospital. Today is very positive to see that the development has started. It's going to create a lot of employment and positivity in Limerick.
But I think the absence of a large private hospital in the Midwest has been a deficiency, and therefore the building of this new state-of-the-art private hospital with 150 beds and lots of ancillary services is going to benefit the region as a whole.
Today is a really significant day for Limerick, and I'm delighted to be here because we need more hospital capacity, whether it's private capacity or public capacity, and we're building that right across the region. So the fruition of today's development is really, really significant, and welcome to the people of Limerick.
So, thank you for that fantastic development. I'd like to bring forward our panel now. So I'll ask Niall to join us up here. We can choose whichever seat you'd like, folks. And also Rob, if you're happy to come back down. And then I'll introduce Jason Kenny and Eoghan and Marina to also join us, and Michael Walsh, Mika, if we pop down. And I'll do little introductions to our panel members. I have to introduce already when we're seated. Now. These are far too comfortable. Are these on? Yep. These always feel far too comfortable as seats to sit in for a session like this, but makes it nice and relaxed, which is what we want. So let me introduce the rest of our panel members. I'll start with Eoghan. Eoghan is a graduate of the MSc in Biomedical Device Materials from the University of Limerick, and she has a Bachelor of Engineering Electronics and Instrumentation from India. She's working currently in Boston Scientific as a research and development engineer, and she's been there, Eoghan, I understand, for the past 4 years, and working with a piezoelectric fluidic team as part of Rob's urology team. So you're very welcome. We're delighted to have you here today. And Jason, to introduce Jason formally, is currently the Interim Chief Strategy Officer of the Bon Secours Health System and CEO of the new hospital. And you've been working with the Bonds Group for the past 6 years, I understand, Jason. And previously you were Chief Operations Officer and Deputy CEO for 7 years at the Cork University Hospital Group. And prior to that, you were the General Manager of Kerry General Hospital. And your background is in procurement, supply chain, and you have a Master's in Healthcare Management. And we're delighted that you've moved to the Limerick region. From pulling people out of Cork to Limerick is always valuable. Now, Mick, I don't think you need a huge amount of introduction. I think many of us in the room do know you, but just to formally introduce you as our Professor of Biomedical Device Engineering and currently serving as the Head of Discipline for Biomedical Engineering in the School of Engineering and also a member of the Bernal Institute, which is based here at the Analog Devices building that we're in here today. And Mick, I think, has been such an exemplar in the collaboration piece and industry focus, but also clinically emphasising the applications of research. So I'm looking forward to today's discussion. So the format we'll take is just, I'll start some opening questions and then I'll open to the floor. So please, if there's someone who's got a burning question, just jump in and we can direct it as needs be. We have about half an hour to 45 minutes. I'm looking at colleagues now at the back from my own office. So, you know I can talk a lot, so please stop me. And when we get into a good conversation, because I'm conscious people do have other things to go to, but it's an important opportunity to talk here as well today. So I suppose, Rob, I'd like to start with you, if that's okay. And you talked a lot about meeting the unmet clinical need and the importance of innovation and thinking about even where there is a current clinical need met, looking at improving the outcomes for that. I suppose thinking about how Boston Scientific takes its approach to innovation and developing new products. Maybe talk a little bit, I suppose, about how you identify that unmet patient need, where patients maybe come in, how Boston thinks about matching the right technologies and people and skills, and all that piece of how it actually works underneath all that you showed us earlier.
Yeah, no, it's really an important piece to how we go about developing products at Boston Scientific. If you take a look at the urology division that we're in, we have about, I would say, 10 different technologies we use to treat disease states within the pelvic floor, all around urology. And what I always tell folks, we're technology agnostic because we want the best treatment for the patient. So that whatever it takes to get there, right? So if you look at some technology companies that develop something, they've basically got a technology, and they're going to go apply it to this disease state, and then when they're done, they got their hammer and they're looking for their next nail, and they're going to go apply it to their next disease state. Boston Scientific takes the approach of the unmet need of the customer. So we do a ton of research, both with our physicians, and in the case, the one thing I probably should have said during our presentation, either the artificial urinary sphincter or the penoprosthesis is one of the few medical devices that the patient actually interacts with. Most of the times, it's just in the hands of the physicians. So we'll go out and do massive amounts of voice of customer to understand what it is. We will go to academic institutions and hospitals and see how are they doing surgery and ask them, well, how does that work? Is that easy? Is that hard? And we try to find what those unmet needs are so that we can take a device such as we're talking about with the inflatable penoprosthesis that's been the gold standard for 50 years and say, what's the next great thing that's going to come for it by looking at those unmet needs?
And just I was conscious listening to you talking about the developments, and I suppose, originally with the design of the product, it's implanted and there's a certain amount of information you get, and that's very patient reported verbally. But now the advent of technology allows much more opportunity for monitoring in real time, and I'm wondering about the disciplines that are needed now as technology and innovations evolve. Where do you see the future needs around different disciplines that will have to come, I suppose, to further inform us of those innovations?
Yeah, absolutely. When you think about, I'll give you an example with the inflatable penoprosthesis. The one thing that physicians do after they put after the surgery is they send the patient home and say, hey, once a day for the next 5 weeks, I want you to inflate the device just a little bit so it breaks up the capsule and that the device sits in there properly. Well, the patient just had surgery on on their scrotum, so it's sore, it's sensitive, and they'll follow up with their patients 5 to 6 weeks later and say, have you been cycling the device? And I'm sure the patient's saying, yeah, but we really don't know if they have or they haven't, right? Because, they've been squeezing it. So this will be the first time we've ever had data to say, hey, are they actually complying with that protocol? But if you think about to those future skill sets, wouldn't it be nice that we don't even have to ask the patient about it, right? We have the right AI in there that says, hey, look, I can detect through our sensors that the patient's laying down. I know that it's nighttime, that because of the pulse of the patient, I know that they're in a sleep mode. I'm going to inflate that device gently while they're sleeping, hold it there for 10 minutes, and get that automatic cycling down. So when you think about it, we're going to need people really in the skill sets of electrical engineering, in the area of software and AI development, and of course the development of all those sensors, you know, the mechanical engineering as well. So it's just such a broad range of skill sets we're going to need, where when you look at somebody like me who was hired at Boston Scientific 26 years ago, they were just looking for a mechanical engineer, right?
It's fantastic. I'm a physiotherapist by background. And I remember when I started working first, patients saying to me, will you know if I've done my exercises at home or not? And of course I told them I would, but I wouldn't. So I mean, the opportunities digital health are bringing to all of our lives. And as you say, for ways of optimising ultimately the patient's life, that's just so fantastic. Thank you, Rob. Eoghan, I might move on to you, and I mean, you have the wonderful position of being able to work with Boston Scientific as an engineer in R&D with Rob's group. Can you talk a little bit about maybe your career journey and how you've kind of arrived and what you see as, I suppose, the exciting elements of being involved with this group and what you think some of the future opportunities will be?
So I'm basically from India. So I did my bachelor's in electronics and instrumentation back in India, and then I moved to Ireland to do my master's in biomedical device materials in UL actually. And then while I was doing my master's project, I was working with Dr. Eric Dalton on the optimization and design of the microfluidics pumps that Eric mentioned before. So that's how I met these guys initially the first time. So they were looking into different pump technology that can be used in the EIPP device. So then I got hired to Boston Scientific. So then my career starts there. So now I've been with Boston for the past 4 years, working in this team to develop this, understand and develop this technology to where we are right now. So, so my day-to-day task in Boston Scientific is mostly around understanding what this piezoelectric pumps and valves can do, you know. So it's basically, we are characterizing the pumps and valves to meet the user needs, that unmet needs that we are trying to implement on the new device. It doesn't stop there. We are trying to understand what is being there out in the market right now. So as Rob mentioned, the aim is 700 devices, the gold standard. But again, like, you know, how can we make a new gold standard, you know? So that's the kind of like detail that we are going into. And I'm trying to understand what are the different use cases when the device is being implanted, you know. After that, what are the use cases? How can we mash that to develop a test method which can simulate that kind of performance back in the lab, so that we know that, like, you know, the device that we are going to launch in the future will meet all that unmet needs. And so that's kind of what I'm working on right now. So it kind of allows me to have conversations with the physicians to understand, like, you know, what are the problems that they face during the surgery right now, and what would they like to change during their surgery. And again, from the patient standpoint of view, like, you know, what would they like to improve? So can we incorporate that into the new technology? Again, then the academic collaborations that we are having right now. So I'm leading one of the projects with Trinity College and Amber. So that's kind of understanding how the device now work after implantation, you know, so understanding the tissue characteristics, because there is very limited knowledge about, like, you know, the tissue properties, and we are trying to understand what the current actual device does. Can we develop a synthetic model by partnering with, like, universities like Limerick, and, like, you know, using that academic knowledge. So that's what kind of my day-to-day job. And I feel like, you know, so working with Boston Scientific, it's kind of giving me this diverse opportunity to improve my leadership skills and kind of improve the networking that I'm getting, not just with the Boston, with the academia and with the physician interaction. So I think it's a great opportunity, and this is kind of every day it's a new challenge, and, like, you know, you're doing something new every day, and it's good for saving the life of the people. So that's all the work that I'm doing right now. And again, I'm very thankful to be back here to represent, like, you know, Boston Scientific and UL, because I did my master's here. So thank you so much. Yeah.
We're delighted to have you, Eoghan. It's obvious, I'm listening to you, the passion and the interest that you have in your role. And I suppose I'm curious, did you anticipate when you started your career that all of that would be such a central part of your role. And what I'm trying to get towards is, were you pulled towards a role like this because meeting the unmet clinical need was to the fore, or did you have that kind of conscious awareness in advance of the role? Does that make sense? Because it sounds that's really, really important, and sounds like it's a real central core part of the role.
So when I was doing my master's project, like, you know, I knew what were they trying to do, like, you know, so I kind of knew before me hired to the product, like, you know, because I was working with Eric on a similar kind of, like, you know, during my master's, so I knew that what I'm going to work on, and, like, you know, so I was ready to be there to understand and, like, you know, to be a part of it. So that makes sense.
Thank you very much. It's great. Now, Jason, you've been listening to colleagues talking, I suppose, about collaboration and the importance of that academic, industry, partner, clinical, patient collaboration. From your perspective in setting up a fabulous new, very welcome hospital close by, what's your sense of where that, you know, valuable collaboration ethos can sit with the Bonds Hospital Group?
Yeah, that's a very good point. And first and foremost, thank you very much again for the invite this afternoon. No, it's fascinating, and the title of today is like idea to patient. The one thing we have is the patients. And with the new hospital and currently in George's Quay with Barrington's, we've over 40,000 patients a year coming through in terms of diagnostics and procedures. In the new hospital, it'll be over 100,000 patients coming through the facility. So from our perspective, we can't operate in a silo. We have to partner with the likes of research and clinical trials and with academia, etc. It's vital, because everyone depends on each other in relation to that, and ultimately the patient's going to benefit from that area. In my current role, and I was the CEO of Barrington's Hospital up till last year, and I have a new role this year in terms of chief strategy as well as the new hospital CEO. It's great being the hospital with a new CEO because there's no patients and no staff. It's not the toughest job in the world at the moment, but anyway, best performing hospital in the group. But so in the strategy role, we're more outward looking. So and it's fascinating at the moment. I just spent the last 3.5 days in up in Kildare, where we had companies from America for electronic health record. So in the Bonds, we are not 100% digitalized. So we're about to embark on the 5 hospitals becoming paperless. And it's going to be a massive, massive change. The EHR itself is the vehicle, but it's digital transformation. But what we're listening to our American colleagues and around the world, those who've done this, and it is beginning to, most hospitals will have to move down this road eventually, is that the data coming from the diagnostics and coming from the interventions is available then for use with external universities or academia, etc., to see what can come from that. So to me, I personally feel we're on the leap of a major technological advancement in healthcare. The likes of the major tech companies are now showing their focus towards healthcare, the Amazons, the Apples, Googles, Microsoft. There's going to be a major change happening in relation to this, and the companies we've met, and we've got consultants from Canada helping us with this project. They said it's never been as busy in the last year and the coming years, with hospitals wanting to make that move to digital transformation. And the data that comes from that is absolutely key. So just on, I mean, with the new hospital, and it looks well on the on the screen, to be fair, but what's slightly missing from that, and not necessarily totally known, is behind that building is a new building called a Clinical Services Hub, where we basically are building our outpatient diagnostics building, not in the hospital. So our outpatient diagnostics is primarily your radiology diagnostics, your MRIs, CTs. We're actually putting a PET CT as well, a SPECT CT, X-ray, the whole lot, CT scanners, cardiac CT. That's for outpatients and protecting the radiology modalities within the hospital for those who need it in the hospital, allowing then those radiology devices in the Clinical Services Hub to be used in a less acute manner and more towards the research area as well as an opportunity. So I'll be looking for, heads up, I'll be looking for opportunities to engage with UL and with whoever, RCSI, etc., in terms of, and to be fair, I've come from Cork. I worked in Cork University Hospital, and I see Professor Burke here in University Hospital Limerick. These are busy Model 4 acute centres that have enough on their plate. And the likes of our hospital, which is more elective in nature, can work in collaboration with that whole network to allow a bit more breathing space to be able to do some of the research, do some of the trials, etc. That's not instead of those major level 4 trauma centres, because they've got a different patient cohort and type that needs to be looked at as well. So I think it's exciting. And what I have noticed, and I like your comment about a Cork man coming to Limerick, still living in Cork, commuting up and down, which waiting for the motorway. Oh God, only knows. Huh? So it's a fantastic opportunity. But in Limerick, I'm not just saying because I'm sitting in front of Limerick people, but I've worked in Kerry, in Cork, Dublin. The openness here is phenomenal. UL, the council, HSE, everybody. And there's a real can-do attitude here. So if that's the raw ingredient, I think anything is possible after that.
Yeah, no, thanks, Jason, and absolutely right. And we already enjoy a very strong collaboration with UHL, and Paul Burke, as you mentioned, is here. And I suppose that wide ecosystem that we have in the region across our industry partners, academic collaborators with ourselves and with TUS, but also the wider ecosystem. And it's wonderful to hear about that, where you see the opportunity for collaboration, but to complement what's here. It's about making sure that ultimately the entire unmet need is addressed. So thank you for that. And yes, we're all waiting for the motorway as well. Yes, indeed. So now, I suppose you're the person who's at the coalface in the hospital doing the clinical work and introducing technology. And there's one particular question I want to ask you before I come to my next question. I'm intrigued from a motor skill and training perspective about your transition. I suppose thinking about, I'm assuming, maybe incorrectly, that some of your early training was in a more traditional surgical training, and now the Da Vinci training and the robotic training came later. But even if it didn't, where's the, I suppose, current training and thinking about motor skill development using consoles? Where is that now? Is that very positioned now in training, and is it more specialist? I'm also curious because we have an esports lab here at the university. I think the first one in Ireland that's situated in Lero, which is our software institute, and that group do a lot on gaming, consoles, esports, and it's a really hot topic. And we're actually there. You have requests from colleagues in surgery in Cork to look at robotics and brain training and motor skill development and reaction times. So it's a fascinating additional area of research that struck me when you were describing how you use robotics in surgery.
Yeah, so is it on? Yeah. So you're correct, 100% correct. When I started my training, it was mainly open surgery that I was taught and learned. And then when I went away, that's when I was only properly exposed to robotic surgery. So to train and to get up to speed on it, it was very structured. So you had to complete a number of modules on the console. So again, a bit like playing a computer game, you had to achieve certain scores to prove that your dexterity was good enough to progress. And you were basically examined. Then the next step was to progress onto porcine models. So we were operating on pigs, anaesthetized pigs, different operations. Again, you were audited, monitored, and a certain score had to be achieved before you could operate on a patient. And then when you were learning how to operate on a patient, it was by module. So you wouldn't be told just take out this man's prostate. The operation is broken down into a series of steps, and you'd start with the easiest step, and not until you were competent at that would you progress on to the next step, and that was the model that was in Australia. And then when I came back here, it was about 4 or 5 years ago, it wasn't as regulated. You did have to submit all your operations, your operative logbook, and it was passed by a robotic committee. But when I started 4 years ago in Beaumont, it was only the urologists that were using the robot in the hospital. So it was me and 2 other surgeons. Now it's urology, it's colorectal, so the bowel surgeons are using it. It's upper GI, so esophageal surgeons. ENT, and it's very hard to get a slot on the robot. And that example, it just emphasizes how surgery is progressing to more and more minimally invasive. I think it's the same here in UHL. Initially it was Sebastius Guerin, I think, who started on it, but now I know that the colorectal surgeons are using it. So it is very important that people who start using the robot on patients, they're able to produce their portfolio to prove that they're actually able to do it instead of just going in. So like a simulation or lab or a porcine lab somewhere somewhere here would be like a great idea. You'd have a lot of takers, definitely.
Yeah, no, it's fascinating. And just from your experience then in the day-to-day, and I suppose more about technology in general, how easy, in inverted commas, like we've listened to Jason talking about the electronic health records and the importance of starting afresh with a whole digital transformation approach. What's your experience as a clinician in the ease, I suppose, the processes, the realities of working with new technology in a hospital environment? You know, do you find it burdensome, or are there things that could ease, or the learnings for us in all of that in this conversation today?
No, the electronic health records, it's going to be a game changer, because if if you have an operation in Beaumont and then you go down to Limerick with a different complaint, there's no record of whatever happened to you in Dublin. So this would be a great way to unify everyone's past medical and surgical history. But with regard to embracing new technology and digitalization, we're actually surprisingly approachable. So like Paul Burke, I heard him say it in Mika's talk, that we see technology on a day-to-day basis, and we see how it's used on patients. So if someone has a new idea in our specialty, they shouldn't be afraid to present it to us, because we'd be able to give a very quick opinion and gauge as to whether it's something that's worth pursuing, or whether it won't develop into something more substantial. So we're more than happy to give an opinion on any kind of technology that might be related to our field, because it's always enjoyable to see it progress and to make it from the bench to the bedside and see it being used in a patient. Yeah.
Thanks, Niall. And that openness, Mick, I'm going to finish with you as a question before I go to the audience. I mean, your track record of looking at, in research I said earlier, that's commercially focused but also clinically focused is exemplary. How, and I suppose it's a self-obvious question, but the ingredients that support that and the openness, you know, if you want to talk a little bit, I suppose, about your experience, what you think has worked well, and again, thinking back to what Jason and others have said here, how do we ensure that we create environments that support and enable that collaboration at a wider level than just PI to PI level?
Oh, this working okay? Yep. Yeah. I think it probably, well, for me, I was lucky in that during my PhD, I had Professor Pierce Grace, who was a consultant vascular surgeon in the regional hospital. So he was kind of a collaborator on my PhD. So he was the first guy I got to see operate, and you know that experience is always kind of stays with you. And then, but you see how open in general, you know, clinicians are to new ideas. So it makes it an awful lot easier to try and work on something like that. So internally, though, it very much depends on the research students and what they want to do. You know, I suppose in the context of what we're talking about today, and like sort of working with Boston Scientific, academic timelines and industrial timelines are two very different things. So we talk in the 4 to 5 year time frame, whereas industries kind of mean usually, you know, it's much more shorter problems that they need solved. And I suppose it's kind of understanding everybody's kind of perspective and what they're coming at how they're coming at the problem from. So what does everybody need to get out of it? And sometimes it's just using skill sets to solve immediate problems, and sometimes it's kind of looking at maybe 4 or 5 year timeline and saying, okay, well, where would we like to be? But generally speaking, we're lucky that everybody that works in this field is all geared towards trying to improve patient outcomes, because that's ultimately could be one of us that's getting the device. And so it does, I wouldn't say focus the mind, but it kind of sobers the thoughts around that. And yeah, so the other thing I suppose is really interesting is that when you work in this kind of area, everybody, you know, is interested, because you're always talking about problems that happen to people, and people you're talking to are people. So, you know, it all gears towards a very positive attitude.
Yeah, you have a very unifying approach in the sense that you're all a common purpose. You know, everything else gets left to one side ultimately, because you're there for a common purpose to help meet an unmet patient need.
Much better answer than my one, by the way.
Not at all, Mick.
I'm an engineer, folks. I'm not a wordsmith.
I wouldn't claim wordsmith either, but no. One other question for you, Mika, and before I sweep around the audience. If anyone has a question, if somebody wants to get a microphone, I can look around. Your view, Mika, and be as honest as you wish to be on this, because I'm genuinely curious to your view on this. What more can we do, particularly from a university perspective, to support that commercialisation piece, the innovation, even, you know, and I know we're doing loads, and there's a lot we have been doing, you know, with entrepreneurship programs, loads of related pieces, which I won't give justice to in a short length of time here today. But are there additional things that you've observed from your networks, international networks, that would really help? And I think an opportunity here today with this group, you know, is it something like environment and the way we set things up? Are there opportunities to have more, just even a scenario like this where we get together? Do you think PhD students could be exposed more or earlier? What are your general thoughts on some things at university level we could do to really you know, move things on and support them?
I think the main piece that's missing, but it's a national thing rather than an institutional thing, but it's the young entrepreneur who's willing to take on the company and be the person, be the head, and because it's a very particular skill set, and because if you can find somebody that succeeded, then you won't really, it's hard to find, or they tend not to want to continue on and do it again because they've done it. And so when we go, if you ever pitch to an angel network or, you know, some VCs or private equity, whatever, most of the people that you're pitching to are successful, but they don't want to be CEOs anymore. They want to be the person that funds it and then gets to the new CEO to do the work. So particularly for the innovation side, it's a whole training piece, but I'm not sure. I mean, it's important to expose PhD students to entrepreneurship. I mean, I teach it to undergraduates, and the idea isn't that they'll go and do their own spin-out. The idea is that they might go into Boston Scientific and do 5 or 6 years and then come up with an idea, and that maybe is in a different space, but that they would have been exposed to things like intellectual property, invention disclosures, patents, all that sort of stuff. So we try to expose that. Undergrad and kind of the master's level, a little bit more in the PhD type thing, but I think people need to get industry experience as well, because it's a very different perspective. And then you're looking at maybe the kind of age profile of maybe late 20s, early 30s, when those people are properly career focused, and if they're brave enough to take on the idea of doing a startup, then it's it's to find those. They're the hard bits to find. But internally, I do think we're fighting the good fight, and we're doing what we can. I mean, I wouldn't say comfortable, but there's a lot of comfort in working at Boston Scientific than being the CEO of a startup, and, you know, all of the drama and the funding and where's the people and employees and all of that stuff. You see it like it's a particular type of person that's able for that.
And remains able for it, as you say.
Yes, and remains able for it, yes.
Thanks, Mika. I've just realised it's 5:10. I'm enjoying the chat, and I'm conscious there may be some questions. Or, you know, if people have to leave, that's totally fine. But if anyone has a question, yes, there's a gentleman here in the middle, and Ray, you might do the microphone. Thank you.
Yeah, it's kind of for Niall and Rob over there, because the two guys with the foreign experience. So what are the kind of differences culturally you'd have seen between Australia and here in this area, and Rob, similarly for the States and here? Or what are we doing right, or what are we doing wrong? Or is it just a main cultural issue between us?
I can go first. You know, I think, you know, one of the things I was kind of overwhelmed with the first time I came to Ireland 5-6 years ago is that there's a real entrepreneurial spirit within Ireland that I don't think you normally get at every place you go in the United States, right? And, you know, I was just thinking about your answer about, you know, how does a person be, you know, innovative and an entrepreneur? And what I would argue is my team here at Boston Scientific is very entrepreneurial, right? And they're doing it within the walls of a large organization. I'm not sure how comfortable we are every day, but I'll accept that. But you know, it's really it's really the entrepreneurial spirit of the people here that I've seen that I think I take away most when I head back to the United States.
Is it about healthcare? Capacity, inpatient bed capacity, access to diagnostics, access to outpatients, electronic health record, standardized. So there seems to be a lot more investment in those structures or resources abroad. I don't know if it's more investment or it seems to be better run, but there was no issues with waiting lists or access to CT scans, MRI scans, looking up scan results. Here it's still pen and paper, and I mean, we hear about bed crises every once or twice a week on the news, but the capacity seemed to be better, or I don't know, more available.
Owen? Yeah, I have another question for for Niall and Rob. It's about tissue engineering, and like Niall, you worked in tissue engineering for your PhD, and you speak about alternatives to buccal mucosa, and there are, if I'm allowed to mention, Cook Medical. I don't know if I am, but they have a tissue engineered product on the market, SIS, and I'm just wondering, what's the clinical opinion of tissue engineered alternatives at the moment, and what's the kind of industrial appetite for developing them or kind of putting them out into the market?
So for tissue engineered grafts, I have to say we're a little bit apprehensive about them because, I mean, they've been around since the 1990s, and tissue engineering has always been built as, the next step in reconstruction and regenerative medicine, and that's how it was framed 25 years ago. That's why I did a PhD in it, but it's still being built as the next thing, the next step, but there's not many tissue engineered grafts that are being put into patients. So there must be a reason for that.
I would argue my answer is it's very similar, right? So I actually did a first-in-man regenerative medicine project in the around 2009, right, where we actually went and implanted cells in an erectile dysfunction patient, and the one thing that we kind of learned along the way, and I think it was the difficult path to get to, was that when you look at regulatory bodies that review these things, right, they are designed to be for like a pacemaker, right? We're going to build 100,000 of the year, so all of the controls, all of the regulations are built about building one device that's good for the many. Now when you talk about tissue engineering, I want to build a single device that's only good for me, but I might need to engineer a different device that's only good for you. I think the regulatory controls and how you do that, it's a really interesting both regulatory model, financial model, and business model for companies like Boston Scientific.
I do want to say about tissue engineering where I went wrong, and I think a lot of people are going wrong, is that, I mean, I tried to do a bladder. People have tried to do lungs. They're too big. Like a little small patch or graft, a centimetre or two for an ischemic segment somewhere in the body to repair that would be an ideal place to perhaps revisit instead of going for something big, like a knee or an organ. I think it probably has a role in starting with something smaller, yeah, and then maybe progressing.
Thank you. First of all, great discussion and great presentations this afternoon. So my name is Katrina Bradley. I work in the research office, and I suppose a question for you, Niall, as well. When you're talking about your peer review and your publications and how important that was to, I suppose, get endorsement, how do you balance that up against protecting your secret sauce without, you obviously have to talk about your method, and I know Mika would have this, I suppose, dilemma, or maybe it's not a dilemma. How do you, I suppose, overcome that if you want to get into the commercialization of your innovation?
I think you have to back your idea and take the opinion that what you've come up with is probably better than what your competitor is going to come up with, and that you're probably 2 years ahead of them. So it's going to take probably a long time for them to catch up with you. But if you are publishing your results and validating them in proper medical journals, you have a much stronger hand to play when you're presenting and looking for investment or trying to get involved with industry, because their first question is show me the data, show me the proof. And if you're able to hand them a peer-reviewed clinical trial, you're much stronger than, you know, say I tried it in my hospital. You can say, well, here, look, this is our data, this is the journal that it's been published in, and that tends to make the process smoother and quicker.
One really quick one, and sorry, Niall, I know it seems like we're picking on you, but they're great answers. So put yourself in dreamer mode. If Boston Scientific or somebody like Boston Scientific could basically meet one unmet patient need, and you can time travel on this one. What's the most amazing thing you'd like to see in your in your immediate space? One-liner is good.
It's in Ireland. It's not just the one thing I'd like to see would be a smoother journey for the patient from their diagnosis to their operation and cure. Because at the moment, if a patient I'm sure you all have relatives that have been diagnosed with something, and there's an awful lot of hoops that you have to jump through to try and get the medical issue remedied, where if there was just a streamlined process that things happened within appropriate timelines, that would probably I'd be happy with that.
Great, thanks. So it's already being done elsewhere. We just need to duplicate it.
It's not perfect. I mean, everyone thinks that the grass is greener in, I don't know, the States, and I'm sure, Rob, you have examples. Everything's not better over there or in Australia. They do some things better than us, but we do a lot of stuff as good, or if not better. A lot of our surgeons in Ireland are very well trained. A lot of the physicians are very well trained. They're as good as, if not better than, a lot of the surgeons that are abroad. So it's not all bad.
That's great. Thanks very much.
Very much. It's been a wonderful chatting to you all. Rob, thank you very much. Ivania, thank you very much. Jason, thank you. Niall, thank you very much. And Micah, thank you very much. And colleagues, thank you very much. We'll close on that note, and thank you all for your participation. Thanks.